embark upon is really driven by our portfolio. I've had the good fortune at Amgen of working on each of the molecules shown on the screen in different capacities, some traditional process development, some as applications in continuous manufacturing. We see this increase in molecular complexity and more recently a momentum shift into these hybrid modalities, whether it be siRNAs or antibody peptide conjugates. This is really driving us to, one, work more collaboratively with our partner functions in biologics drug substance, but also think about how we innovate our manufacturing approaches to accommodate these new types of modalities. So I'll be brief so we can get into the more informal conversational aspects of this session. But I did want to, again, just baseline how within drug substance at Amgen we think about application of continuous manufacturing. So in my mind, I think about CM falling into two primary buckets. The first is flow chemistry as an enabling technology. That is, flow chemistry is the only way to make this molecule. This can be applied to a single unit operation, a reaction. It could be the aqueous workup, reducing a bottleneck in manufacturing. It could be a crystallization for impurity control. Either way, a really focused effort using flow chemistry to enable a chemical transformation. The second bucket is what we call next-gen integrated and hybrid processes. This is where we're looking at a more holistic view of a chemical entity, thinking how can we integrate unit operations and really streamline the overall process. And if we think about the finance behind these two topics, and this is what we'll get into today, really flow chemistry, in my opinion, the first bucket, doesn't really have primary financial drivers. It's, do you want to make this molecule or not? Flow chemistry is the way to do it. When you think about applications of fully integrated processes, that takes a much larger development team, longer timelines, more infrastructure, both in the development lab, but also the commercial manufacturing facility. And that's where we really need to start to build that compelling business case. So let me just show a couple of brief examples of each, then we'll segue into the conversation. So flow chemistry as an enabling technology is just amazingly broad in its application. That is, we can use it for, in the preclinical space, reaching back into therapeutic discovery, all the way through commercial. Apologies for the formatting on the bottom there. We can also apply this for raw materials, all the way through the drug substance itself. We had a few recent applications highlighting this for the molecule AMG 397, one of the most complex traditional small molecules in our portfolio, about 50 plus synthetic steps to manufacture this. And what's really exciting is that we implemented flow chemistry for step number 1 and step number 50. And so step number 1, we developed a photochemical flow process, so a flow 2+2 photocycle addition to make this key raw material. We're able to achieve throughputs of over 5 kilograms per day using a large 365 nanometer length skid. And then a few steps later, we applied flow chemistry to make the final API. So what's on paper, a really trivial methylation, but in practice, a really, really challenging transformation. Actually, as you allow this anion to sit in the reaction mixture, you see agglomeration, you can see this by NMR and a few other analytical methods that really reduces the nucleophilicity of this tertiary alcohol and eventually results in reaction stalling and impurity formation. So enabling really fast mixing, really efficient heat transfer allowed us to overcome that challenge. I'll move forward so we can get to the conversation here. But just one example, which will kick off some of the financial aspects, how do we apply fully integrated next generation and hybrid processing? We did this most recently with Tesla. I was chatting with some folks from Celgene, now BMS, yesterday in the network session. You might be familiar with this drug substance manufacturing route. It's amazingly efficient. But we saw still opportunities for application of continuous manufacturing to continue reducing cost of goods, but also enable internalization of the supply chain for this important asset. And so this wasn't just a direct translation of the existing chemical process to CM, it took a bit of finesse. And so again, pulling from this toolbox of platforms that we have, reaction extraction, distillation, crystallization, and isolation, and mapping that on the existing batch commercial process with some additional process innovation, we're able to incredibly intensify the process. So from 9 operations down to 3, and then fully integrate those in a continuous manner. Really the key driver for this, again, was reduced cost of goods, improved business case, and internalization of the supply chain. The key chemistry innovation here was really quite simple. It was just finding a solvent within which all the starting materials, intermediates, products were soluble. That turned out to be DMSO. And then through the simple addition of water as an anti-solvent, you can crystallize out your API, perform a continuous crystallization, continuous isolation, and a high-level process flow diagram is shown here for that. Your 2 starting materials, anhydride aminosulfone, mixed in a PFR at 130 degrees. DMSO, as we know, is thermally unstable at high temperatures, but because we have incredibly precise control over reaction temperature and reaction time, plus a number of additional engineering controls to ensure safety, we felt comfortable with this process. That then telescopes directly into the crystallization where water is added, and then onto a parallel centrifuge system. The filtration and drying of this particular API is incredibly efficient, and so parallel centrifuges worked quite well here. And it also provided the added benefit of just discharging directly into drums. So again, these next generation hybrid plays really require tremendous infrastructure and investment for development and into the commercial manufacturing space. I'm showing on the top left here 2 of our kind of walk-in fume hood skids that we use in the development lab, both for development and process characterization. On the top right, I'm showing the internal facility within which this will exist. This facility was kind enough to give their closet to us, but even with that small space, we predict we can make about 5 to 10 metric tons per year. And then I'm showing just a mock-up of the actual skid here, and there's a gentleman inside there, so you get a sense of size. And finally, we work with external equipment partners. This is not, as Olivier showed, a very flexible skid, but because this is dedicated to a single commercial asset with multi-billion-dollar revenue, we made the business decision to make a fixed asset for this particular molecule. And so going left to right, I'm showing the pump section of the skid. The 2 gentlemen in the middle are next to the PFR, which is this coiled tube and a jacket reactor with a number of pre-coolers next to it, or heaters, I should say. In the top right, that's showing the PFR on the left, and then the surge vessels on the right, and then the bottom right then is the MSMPRs, which would then feed into the parallel centrifuge system. So again, juxtaposing the 2 approaches, flow chemistry, which is just absolutely required to make molecules, versus fully integrated processing, which requires significant internal investment, and for Amgen has a number of compelling business drivers. So that's the context. With that, I pulled out the 3 primary focus areas, which I think we will have additional conversations on. So.
Thank you, Matthew. And what Matthew just put up, this is what the input, and in order, the different companies have different drivers, the challenges, innovator, manufacturer, biotech, etc. What Matthew and I and the other scientists had discussion in preparation for this is really organizing their thoughts on what the critical questions are. And we're hoping that what Matt and the others will discuss will impact you and help you understand what's needed. So the first question, could you talk about the stakeholders and groups at Amgen that are needed to be part of the continuous manufacturing discussion and implementation? For example, upper management, IT, finance, research, process development, QA, QC, regulatory, manufacturing, supply chain, logistics. Are there others? So this is a major cultural shift. How is the continuous manufacturing plan communicated? How do you get buy-on to ensure success? And as we think about it, we go back to what Judy talked about yesterday. in having the right mindset, the right personnel, because without that, there's no execution. But Matthew?
Yeah, let me provide a few thoughts, and I think it'd be most valuable to do that in the context of some of the different case studies I showed here. And then I really want to make sure there's some dialogue here with the audience as well, right? I'm coming from an innovator company, small molecule drug substance, but we have a diversity of folks in the room here, so hopefully we get some different opinions. I also got the question in the networking session, can we actually provide some details, numbers, actual hurdles, right, instead of talking in generalities? And so I will do my best, but if I'm not giving enough detail, please just let me know. I'll see what I can do. So again, just coming back to the overall approach here and how we get stakeholder engagement. What I shared at the start is when we think about flow chemistry as an enabling technology, it really doesn't require a high level of stakeholder support. What really happens at Amgen is that we go into the lab, we do our route scouting exercises, and we just innovate as process chemists, chemical engineers, and analytical chemists. In these specific examples, the top right, that was really driven by process need. We made a 10 kilogram batch, impurity levels were rising, and we're actually really concerned that we're not going to be able to control a particular impurity as we scale further. It became apparent immediately that continuous manufacturing was one lever to help solve that challenge. And so that idea came from the lab, process chemists. But then, because we didn't have the internal capability at that time, a few years ago, to develop that process, build the equipment, and execute, that's when we started to reach out to external partners, CMOs, CROs. We worked with a particular CMO to help develop the process and design the equipment, and then we worked with a different CMO to actually execute that manufacture, and that was a preferred external supplier. So again, really driven by process need there. I don't think there's a tremendous amount of stakeholder engagement or approval required. Now, just contrasting that with the Otezla example, this one really came from the top down. We've had these conversations at different groups. At a high level, let's say SVP level, there was a real desire to internalize this asset, which came back down to me. What would be the way to do that? Well, we can't make this at the existing commercial facility. Again, I'm showing a very blurry picture here, so feel free to ask for details. This manufacturing facility within the Amgen walls has the largest reactor of about 1,000 liters. You can't make 10 metric tons of Otezla in that facility by traditional processing. The only way to do that would be through a continuous process, which then required innovation on the chemistry, work with chemical engineers to characterize the equipment required to execute that chemistry, work with analytical chemistry to ensure that we understand new impurity profiles. We're using new solvents in an API forming step. What's the clearance of those solvents? Working with regulatory to refile this existing commercial batch process, quality, so on and so forth. Because this was going to an internal facility, of course, a full-blown business case was developed. This was going overseas, and so through the tax considerations, so on and so forth, was really quite important. So this one took everybody to come together. So that's Innovator, DS, CM. But I'm curious if others, have other thoughts or additional questions to that particular topic.
From the innovator companies that presented yesterday, any comments from the Amgen approach? Is yours any different, or do you have anything that you would like to add? And that, if anyone will respond to that, and we'll follow it up with questions from the audience.
I'm going to ask a question on the audience, a specific one. So CMOs investing in continuous manufacturing. I've had conversations with a lot of you in this room on that. I'm curious if that's driven by a company like Amgen, or you are perceiving that there's a very strong business driver to do that independently, or a combination of both. I'm looking at specific people to answer that, but that could go to anybody.
From a CMO perspective, we've done continuous processing for a while, mostly to deal with energetic chemistry. And so I agree with one of your slides. It's really an enabling technology. So it's a way to get into chemistries that you would not be able to do batch-wise. And so we've continued to build on that, and doing projects with an Amgen or other innovator companies in this space is just helped us build on this technology. I don't know if you want to elaborate.
Well, I'm biased because I'm a chemical engineer, and as a chemical engineer, continuous process is always better than batch in general, for many reasons. But like Bill said, we have done continuous process for a long time. We do on a routine basis continuous separations, and so we have all the understanding in-house from all the different groups internally. So QC, QA, they know how to handle those kind of project or technologies. So moving towards full continuous is something that's easier for us because we're already in there. And like Bill said, companies coming to us with the need for continuous steps helps us develop more our technology and being able to offer a platform that will eventually make scale up more flexible, easy, and reduce that timeline. But no, this is an excellent example that you provided there on the benefit of the continuous. So hopefully we'll see more hybrid processes, and I think that's where we're all going to go eventually.
Matt, I'd actually like to ask you if you could just elaborate again on the drivers why Amgen felt that Tesla was the right candidate to do a integrated CM project.
Yeah, absolutely, and I'll try to be as specific as I can, and I'll speak to it from a few different angles. Selfishly, from a chemistry perspective, this one's really easy. I'm just being very, very transparent here, right? And so if you look at how the supply chain is set up, you would actually get these, depending where you are in the world, starting materials, raw materials, or regulatory starting materials from another supplier. In theory, those starting materials would then be shipped to our manufacturing site. We would need to execute one chemical step to make our API, and then there would be a final batch crystallization, which intercepts the existing filing. And so the only continuous portions of this would be this penultimate chemical step. And just given that it's a condensation, it's a relatively straightforward step. We have a very good understanding of the impurity profile, so on and so forth. And so with relatively minimum investment in the development space, we can very quickly achieve proof of concept and say that, yes, we can do this. We also have existing equipment to support the development of this, and we saw value in investing in these particular skids in the manufacturing space because we perceive them to be very flexible long-term. PFR, MSMPRs, and a few centrifuges, which are generally needed across any chemical operation. And so from a technical perspective, it made a ton of sense, and there was a very strong upward push, I would say, from my group to say, oh, Tesla is the right application. The other desire there was to make sure that we had enough runway to achieve some return on our investment. And so, if I'm not mistaken, the end of our exclusivity on this particular asset is, let's say, 2028. This is public knowledge. And therefore, at the time when we started this, there was enough time to develop a process, build a skid, implement it, and gain some returns on that particular investment. So that's kind of supply chain technical considerations and also some of the financial piece. The other very strong driver was to increase utilization of our internal manufacturing facility. So I showed it's a very small facility. It's dedicated to a single asset at the moment. And we want to make sure that we have some continuity and utilization of that particular site. And so carving out a small little corner of that to make 10 metric tons of Tesla, let's say, seemed to make a lot of sense and brought tremendous utilization to that facility. So supply chain, making more Tesla for the world. Generally positive business case. I can provide some thoughts there as well. It doesn't have to be, $100 million, $1 billion NPV. To get all this into the facility to implement this process was, what I would call a neutral NPV, plus or minus $10 million, let's say, trying to provide some details. But what it did was it allowed us to go through the entire process of developing a process, designing a skid, actually fabricating that skid, implementing it, and going through the regulatory hoops of bringing on a new continuous manufacturing process. So the timing made sense for Tesla, the chemical challenges associated with it made sense, and the availability within the existing internal manufacturing sites seemed to make sense. And so everything sort of aligned for Tesla.
Please, yes. I have a follow-up question. In your assessment for efficiency and return on investment, what was your experience with changing the regulatory aspect of a second generation process, especially as this is for the API, as we heard probably for the drug product, second generation process might be more challenging.
Great question, and this is still an evolving story. I can be completely transparent. But there has been engagement with the FDA. There has been discussions around ETP, et cetera, and maybe I won't give much more details on that in this forum here. But generally speaking, the response has been very positive for bringing on a new, more efficient process that, again, intercepts the final batch crystallization, delivers the same particle size distribution, delivers against all of the same other release specifications, right? So I think that's an important part of our strategy and one that we use for all of our lifecycle management approaches. That is, perhaps translate some of the batch inefficiencies to CM efficiencies or upstream chemical steps, but then intercept the existing commercial filing for the final batch crystallization. And that, I think, helps us out a little bit. I think one of the challenges that we have, not a challenge, but an opportunity that we have perhaps, is a advanced control strategy. So we've talked a bit about control strategy over the past day or so, and how PAT can be used in development and in commercialization to different extents. Our intent here is to have no analytical testing across this process. We have someone unmuted.
Online staff, please mute your phones. We can hear a lot of static. Thank you.
So just to come back to the control strategy, I think that's where we'll see a little bit of resistance, but we are very confident in this approach based on the hundreds and hundreds of process characterization experiments that we performed in corresponding models, that if we achieve adherence to our set points, our parameters, whether it be PFR temperature, flow rates, et cetera, that we do not need to test throughout this entire process, that what is coming into that drum is adhering to our product quality requirements. And so we'll have additional conversations on that topic, I'm sure.
So, in your examples here, I mean, you're talking about taking a batch process and making it continuous once it's launched, right? So a second generation approach, other than like the 397 where you have a 2+2, which is going to be difficult to do batch-wise. What is your philosophy on starting to introduce CM earlier in clinical trials, or does speed preclude that?
Excellent question. And I think I can tie this back to financials to some degree, but I'll answer it directly. The easiest avenue for exploration for us at this point has been lifecycle management. That is, we have an existing supply chain that we can always fall back on. And so as maybe technical development fails, or a business case falls off a cliff, or any other reason why you might stop implementation, you always have an exit standard process. So that's been the approach that we've taken for all of our commercial assets to allow us to develop skids, to develop capabilities, et cetera, in the context of that, I'll call it safe place. What's not so safe is first intent continuous manufacturing. And a lot of companies have done this and have been successful. What we don't have is an existing manufacturing site which could accommodate that at the moment. And so that would require us to work with you or others to enable those first deliveries to support clinical trials. And so there's a tremendous opportunity there. We have not embarked on that path yet. We've thought about it for some assets, but to your point, failure rate's very high early on. For an integrated process, as I mentioned, it takes a lot more infrastructure and resourcing to develop that process as first intent. I don't think it's going to delay the clinical study. I'll never be on critical path. I'll always deliver the DS, but it's going to take a lot of resources that could otherwise be worked on other assets within our portfolio. So that's been the challenge. If there were a facility that was plug and play, that would change things, I think. That could be the CMO space, it could be the Amgen network. But my thoughts is we're maybe not there yet where I could just ideate a process and drop it into a facility and have it manufactured in a couple months. For batch, yes, we can do that very, very easily. So I think that's the struggle and why we haven't taken that leap yet.
Can you please use the mic?
I can repeat the question. At what point would you do both CM and batch early on? I think this came up in the conversations yesterday, if I'm not mistaken. Do you have to recreate the wheel to do a CM process? Not necessarily. If you're developing a batch process in parallel, I think you can leverage a lot of those learnings. So it's not going to be double the team, but pretty close, I think. And again, just because we operate in a relatively lean structure, at least within my group, it'd be hard to justify using those resources when they could be developing a process for a different clinical asset.
Another question.
Just one question. What is your strategy for future products? Like, do you have a criteria on which you would say we'll pick these steps for flow, or like while apremilast had a specific set of reasons? What do you have a strategy that you're going to follow for the next set of molecules we have in the pipeline?
Yeah, so I think that's very much evolving, and that's a tremendous question. Because in theory, we would have this beautiful flow chart, and it would just be so obvious. You see a chemical entity, you have the route scouting meeting, and you say, through your flow chart, this is flow, this is batch, this is hybrid, right? But I think it is more complex, because what needs to happen, and which is very much happening in the space now, is you need to build in all of those considerations into your route scouting exercise. And so I'm thinking from, as a molecule enters my group in phase 2 studies, right, we have an opportunity to rethink the entire process, route scouting, commercial process development, process characterization. It's at that route scouting phase where we would not throw away what was done previously. We definitely want to leverage that, but we have the opportunity to completely redesign the route. Now, if the people in that route scouting meeting don't have flow experience or continuous manufacturing experience, they might not be thinking that a 2+2 flow cycle addition is even feasible. So we need to make sure that we build those capabilities very early into all of our ideation meetings. I think on the flip side of that is as we start to or continue to in-license assets, that's our opportunity perhaps to think very clearly, not just from a technical perspective, but also supply chain and financial perspective. What is our overarching strategy? Do we always want to in-license a molecule, translate to CM, internalize that manufacturing network? If you ask me right now, I'd say no, not for every single one, but there's probably some, like the one I'm shared, where it makes a lot of sense. Yeah.
Perfect entry for my question, which was, it sounded like for Otezla, with the management life cycle. It was the long financial runway. It wasn't, you have time left on a patent. The desire had a need in a plant to fill a location, and so there, like you said, there's a supply chain there, and then there was the opportunity to improve the process through intensification, which is going to be some chemistry dependent. It sounds like without all three of those, this wouldn't have happened. So for you as a innovator, it's going to be a very molecule by molecule type decision in that type of thing, in that type of way, right?
Well said. It's absolutely. As I was speaking to earlier, it really comes down to what internal resources do we have at that moment of time to actually support an endeavor like this. Loss of exclusivity, let's say, is 2028, it's 2024, right? So we need to continue moving here, but all of those folks that were dedicated to that project were not working on a different project. We have more than one commercial asset, right? In theory, to your question earlier, any one of those could have been chosen for this. We have more that will be licensed in the future, I'm sure of it. And so I think what we need to continue to do is build in those resources proactively such that when the next opportunity comes, we can jump on it immediately. But it's not just process chemists, right? To your question half an hour ago, it takes a village, right? It takes many different functions to enable this. And one other roadblock that we run into, right, is we can develop the best process in the world. I think in some cases we've done that for some of our assets. But if the other functions don't have bandwidth or resourcing to support implementation of that, then you're sort of hosed, right? So at what executive level do you need sponsorship from to make sure that everybody's aligned early on, and it's not just a few process chemists kind of working on it?
Another question?
Yes, we have one from someone online. They're going to unmute.
Hi everyone, I have one question regarding the validation strategy. Part of the regulatory filing, could you please elaborate more on the validation strategy? Did you perform like a full process validation or like CPV, continuous process verification? Given the fact that the validation work take time and also it can be higher in terms of cost, would you file within the regulatory dossier a scaled down model and later on you will provide the validation data, or you file already the full package, like the validation within the dossier to get the approval in a reasonable time frame? Thank you.
Tremendous question. Let me start with an answer, and if I don't touch on those topics, please just chime back in. Again, tying this back to financial considerations, you're absolutely right. This was a constant point of discussion with executive leadership, looking at numbers, right? So, in my very strong opinion, it's probably in our best interest when you receive a skid like this directly from a vendor to run solvent through it and maybe do a couple of test runs. But that costs money and it takes time. And so there is very strong pushback to receive this equipment and start PPQ. I don't see how that's technically feasible, right? So we need to build in the time and the funding to support full understanding, characterization of this equipment, et cetera. Solvent runs, residence time distribution studies to ensure that our scale down models were appropriate. Then engineering runs like plural, and then validation. So to answer your question directly, that was a topic of significant debate. Our intent is to validate on this equipment using a very traditional approach. We're taking a very conservative approach here. So this would be 3 full runs on this commercial equipment. Those runs would be informed very much by all the work that we did in the development lab. We characterized the development lab equipment such that we had strong confidence that those studies are representative. But of course, as we go through validation, I think this came up yesterday as well. In those validation runs, I think we would intend to stress this equipment, at the edges of those normal operating ranges, see how the equipment responds to those disturbances, etc. There's of course also a lot of discussion around, is that actually required to file a continuous process? Do you need to impart, intentional disturbances? We have a good sense from our scaled down equipment what our tolerable time for disturbances are for each of the parameters that I mentioned, because this is based on parametric control. That's our intention for the control strategy. It would probably be important that we test this on the actual commercial equipment. And so the plan is to build that into the studies. So again, 3 full batches on the commercial equipment, but of course in S26 leveraging all of the data from the small scale runs.
Oh,
you talked about what is important for Amgen. How do you see for the industry in general the evolution, and how would the financial barriers be likely to be lowered in the future? I can imagine one of the examples could be vendors producing skits that are then standardized and are cataloged, and that it would be easier for companies to just pick and choose, connect them together, standardization of interfaces. Another thing could be digital twins that help to prepare for that. What are your thoughts on what can really help the industry to lower the financial barrier for this?
I'll give a few quick thoughts, and I want to ask the rest of the audience to chime in as well. From my perspective, every conference I go to, every workshop I take part in, there's always a topic: how can we standardize equipment? And we see this in some of the slides from yesterday. Every company that presented has a PFR, but every PFR you see is different. It could be a tube in some sort of heat exchanger. It could be PFA in a big bath. It could be this thing that I showed, so on and so forth. So even the most simple reactor design, a tube, is different at every company. The pump selections, what's actually pumping those fluids, is different at every single company, which I find very interesting. Through IQ as an example, a consortium that's supposed to bring us all together, we've had those conversations, and we still come to the point where, well, I actually sort of like this one, I sort of like this one. If I need to handle solids, it'll be a peristaltic pump. If I need pulseless flow, it'll be X, Y, and Z. So the theory of equipment standardization, I think, is amazing. The implementation, I think, has been significantly challenging. And that's just the physical piece of equipment. Once you think about the automation behind that, every company has its own platform. Built to be, I think we heard of some others at Lilly and otherwise. And so getting all those to integrate across all the companies is really quite challenging. I think it would be amazing, but it's not the current state. Would that bring down costs? I don't know. Is it a single partner making all this equipment and they have a monopoly? I don't know exactly the implications of that, right? But it would make it easier, I think, for me to tech transfer processes, because a lot of our processes are executed externally. If my development lab had the same exact equipment as the CMO, CRO, that would be seamless. And then if there's a seamless scale up to the manufacturing plant using the same equipment platform, that would be beautiful, right? So I think we have that internally. I think Lilly has that internally, and AMBAC is building this internally, so on and so forth. But each one's a little bit different. That's just my thought. I'm curious if Hank and others, thoughts on equipment standardization and others.
Marcel, I think. Sorry. Thanks. Yes, as a system integrator, I think you have to differentiate already from, are you equipment manufacturer, just have a reactor, or do you integrate with all the automation, etc. more? And I fully agree. What we have seen is all different. So there's no standardization. And if already you could look at standardization, it would be for certain chemistries. Take, for example, organic metallics. There you could probably standardize a skid to a certain level with a certain throughput, etc. more. But I think that would be the route to go, is start with the chemistry then, and there could be some standardization in skits for certain chemistries, not for what we have seen, but with all the customers we built now for, it's all different. Different pumps, different this, different that, and they all, in the end, look rather simple when we are the same if you build them, but they are totally different.
I think the only thing from my perspective that has seen a lot of initiative to standardization is from the control systems perspective. And this is evidenced by Emerson as well as Siemens. So, I think from what I've been hearing from a lot of European companies, it's relatively easy to exchange systems in a plug-and-play fashion, but what is really challenging is trying to integrate that into your plant-wide control system. So there's been a lot of work on the control aspect of standardizing these with certain, I wouldn't say regulatory standards, but I would say some internal industry standards. So there is a lot of success in that, on that front.
Thanks, Stephen. Maybe I can ask I was just going to ask real quick a question since you have the microphone. We were having this conversation yesterday, and I think it's really interesting as it pertains to financial. It's what would incentivize you or a company similar to actually develop equipment? Is it I come to you with a process need, or you see a gap in the field and you're incentivized to develop that on your own time, let's say, and then release it to the wolves?
So I think having an existing revenue stream or customer would be really great or a client to really come to us with a problem. Also, I think one thing that we've been successful as a company is being able to understand where there are huge gaps in technological solutions. I think every company that practices flow chemistry has had some exposure to the flow chemistry, even to some continuous crystallization on a drug product. A lot of companies have drug product continuous processing initiatives, but really thinking about what it takes to integrate these. Eli Lilly's had some great success at that. Amgen, GSK, even Novartis to some extent. But I think one of the really challenging things that hasn't been seen are things that are purification steps beyond just extraction. So filtration, drying, polish filtration, and on the biologic side, tangential flow filtration, or more automated ways of doing chromatography. So I think that there's a lot of opportunity for our CDMOs to really develop these, and it could be a joint project to develop that in a non-competitive way with industry, innovators, and then really, then have that asset that's already de-risked with maybe a model compound to really then take on additional projects. So trying to find, I would say, unmet need from an industrial standpoint, and then being able to work together on that.
Well, if we talk about standardization, even in batch, there's not really a standardization, because when you bring a project to a CDMO, you still spend a few weeks at a CDMO to do the tech transfer from your bench batch process to the CDMO bench equipment. And then it falls into the CDMO to now they approve the process at small scale to take it to their facility internally. So that standardization is inside of the CDMO, right? So every process come in, it doesn't matter if it's continuous or batch, the CDMO is going to spend time to understand the process and see what are the knob to turn to put it in the plant and make the 10 metric tons, 20 metric tons that you need, right? So I think for the continuous process, there's still a lot of uncertainties unknown about technology, but from a chemical engineering perspective, we need to define better process in terms of not just the kinetics, but also the mass transfer, the heat transfer. And when you buy a distillation column, you need to know the number of plates, the heat exchange, the surface area, right? There's some key parameters that you need to define. Same thing for the reactor. You need to know your Reynolds number, your turbulent flow. Very, very frequently I ask what's the Reynolds number in there, and I get deers in the headline. Look, right, nobody calculates that, but it's this basic number you need to know when you do PFR. But nobody does that. in this pharmaceutical environment, right? So if we better understand the process, then we can bring it to the CDMO, go look at that process, look at what are the knobs we need to turn to put in our equipment, in our facility, and then it's our job internally to have our standard equipment, so it's easier to adapt the process to our facility, like the way we do in batch. It's not going to be much different.
An analogy, yeah.
But I think also there might be an opportunity to, I think it's called the ETC in Philadelphia, who companies line up to, they have a certain subject or objective. I think for flow solutions, that could be a great opportunity to join forces there as the pharmaceutical world and take certain examples of certain chemistries and tries to standardize this. That would introduce continuous manufacturing and technologies much faster, I think. But you have then to open up as companies, also as suppliers, let's say, of equipment.
Yeah, it's a really good point. Leveraging those existing consortia to support some of this is really, really valuable. I'm familiar with many of those work streams. More recently, there's electrochemistry, photochemistry, a number of other things that would be amenable to CM, right? So, yeah. Hi there.
So, yeah, just to bring out some of the context here, that this is a very drug substance oriented talk, which is excellent, but I see this as like, it's a spectrum where if we go, we don't have any packaging people in the room that I know of, but packaging is highly standardized, and they have like PackML, and they go into automation machines, then you get the drug product, and we have a greater ability to call unit operations and standardize, because we're doing mostly just mechanical transformations and not chemical. But then as you get into drug substance, it's like an explosion of complexity, which would make it all that much harder to standardize something that would work for a large number of products and chemistries.
Thanks, everyone. It's been a great discussion, but we need to move on to the second question. In this one, this was voted on as number 2 from you. Lack of business cases showing that continuous manufacturing is more cost effective than batch, especially for generics where margins are slim. So the questions, Matt, has Amgen built new small molecule facilities, or have they repurposed existing facilities? And from a capital investment perspective, who and how is this determined?
On a few of these different points here, we covered some of it. You know, we have a single small molecule manufacturing facility at Amgen. We're looking to expand the capabilities of that facility, and that's a very overarching driver for implementation of CM. Is CM more cost effective than batch? I think that's interesting, right? So you can come at this from a couple different angles. Let me just share my experience real quick. When we develop a process like this for Otezla, which shows a reduced cost of goods, when we show 2+2 photocycle addition, which enables new chemistry, eliminating 5 previous synthetic steps, etc. When you present that, folks within Amgen will ask, perfect, do that for this one, reduce cost of goods, make more molecules, right? But you can't necessarily do that. So is CM always better for every asset? It's hard to say because they're so individual. I think the other challenge that I have personally, I'm curious others' thoughts, is that it's very hard to compare directly a CM process to the batch process. Anytime we've developed a CM process, it's based on learnings from batch development, minor process changes that could, in theory, also be applied to the batch process, but enables intensification of the continuous process. And so it's never an apples to apples comparison. You just come out, you provide a stakeholder presentation, and CM just looks better because that's what we're trying to push for, right? In the Otezla example, could you apply those same changes to the Otezla batch process? Absolutely. I think the main issue there would be high temperature DMSO reaction. You just would not do that in a large batch reactor. But that's just one example where all the intensification we learned through developing a CM process could, in theory, be back integrated into the batch process. So is CM more cost effective than batch? I don't know, thoughts?
So once you start getting into the cost effectiveness, this is really about the business case, right? You said something very important about Tesla, which was your runway, how long you had. You guys had 5-6 years of guarantee, right? And this is where it comes into how our supply chain is structured and how companies work. That since we do have batch, we're very flexible. We tend to do a bunch of manufacturing of molecules at a bunch of different facilities from a global supply chain perspective, particularly in generics. You might get APIs from 5 different plants or something like that. If you were looking at an asset for its full life cycle and looking at what was best for just that asset, you said, okay, we're making, product A. This product A is probably going to be taken in volume for 30 years. There's no doubt in my mind that take away all of the different things, you would want to maybe get it out quick. Maybe that's great with batch, but then you would then go to improve your efficiency over time, and maybe eventually, like a chemical plant, you might have one plant with recycle loops and everything where you're really maximizing your yield. But our business processes, our organizations, they don't support that. We go from innovators to generics right in the middle of the thing, and then the generics is then competing with other generics, and they're all, trying to get down the volume. So so CM for a high value thing could always be made to be better, but you have to have business certainty of a 15, 20, 25-year plan, which we don't. We tend to work on this 1 year, 2 year type cycle, right?
I can provide some perspective on the costs in development. So really not doing CM, by doing CM, we don't have to scale up, and that's one of the biggest drivers, and because of that, significant cost savings in development. So I have a question on the commercial side. I mean, how, the perceived advantages that we have heard of are really, reduced waste, less number of operators to operate CM plants. So what does, in terms of when we look at drug product, even for batch drug product made using batch processes, the yields are really, really good. In fact, for smaller batch sizes, continuous processes struggle to get good yield. So that's really not a driver for going with CM in commercial manufacturing. So what is the experience with companies who have implemented CM in commercial for drug product? Are they really realizing the cost savings that they originally envisioned with less number of operators, efficiencies, time savings in cycle time, cleaning and such? What's the experience? Drug product, you can start with drug product, and maybe there are similar analogies in drug substance we'd like to hear.
Eric or others, please, on the T consent.
Yeah, so I think it's this is about the potential of the technology versus the reality of the technology, and I think it would depend on where it was implemented and how. Certainly a continuous, from a CDMO perspective, when I was with Patheon, a large aspect of the program was cost reduction over long term, and that line was designed and built out now to be efficient, run a lot, high utilization, minimum operations. That is not like most of the lines that are in the industry today, because most of the lines in the industry today are built to launch products. And so they're built to have, fire drill, lots of hands-on, get the product out the door, and have as little risk in the control strategy from a filing. So if agency asks for it, give it, give more control, more assurance of quality. So it's actually the reverse with a lot of the lines that are out today, because they're built for rapid launch. They tend to be very expensive in operation. That's not the technology's fault. That's the business case that was followed in order to make that happen and enable that quick launch, where there is a massive business benefit to saving 2 or 3 months on a breakthrough therapy. Massive, pays for the whole program 3 times over.
Well, we have just over 3 minutes left. So Matt, I'll turn the floor to you to kind of wrap up and summarize Any additional thoughts?
But this has been a lot of fun. I'm happy to get off the stage in about 3 minutes here, or less, if I can keep it brief. But thanks for the audience interaction. We have a few more of these coming up, which would be great. CM for me has been just an amazing experience. That is developing processes, implementing processes, tech transferring processes, working with many of you in this room through industry consortia, through tech transferring processes, through building skills, knowledge, etc. We're at this amazing point now, right, where we can have these questions, these dialogues, and there's actually enough data in the room to actually drive some change and answer these questions. But there is still so much outstanding, and there are still so many questions. We didn't touch on this last one directly, but it sort of came up in a number of questions here. Can you go all in developing CM processes for all of these assets that are either going to fail in the clinical studies or have limited lifetimes, exclusivity for the innovators? I've had personally, just to close out this one, many failed business cases because demand dropped post-commercialization. We had a developed process, but the MBV just falls off a cliff, right? And that seems to happen more often than not, unfortunately. So how can we overcome that? One, if you never have to compare a CM process to a batch process that is as first intent, your CM, to your point, then build that capability early on, and that is just the way you manufacture. But what does that require? That's internal knowledge, SMEs, infrastructure, development lab, commercial, clinical as well, to make that happen. And so at least at Amgen, I think we're very actively working towards that. I know many companies in this room have achieved that to various degrees, and it's really inspirational to see. So I think the future is bright. From a financial perspective, I should have given this caveat very early on. I'm not in business development. I'm not in a finance group. Just mute yourself if you could. But I think together as a team, we can continue to learn, to work through financial, regulatory, technical hurdles, and I'm looking forward to it. So thanks, Ernie, for hosting. Thanks, Dennis, for the invite. Others, appreciate it. No more questions.
So since we are on the financial consideration topic, right? So, companies such as BMS and many others, right? The pipelines involve quite diverse set of molecules. There could be, 25% high potent, 1 or 2 high volume peptides and whatnot, right? How do you make a business case for a technology for which the expected assets are going to be, let's say, 5-7 years down the line, and by the time you get there, those assets fall off, really? So, I'm sure many of you must have gone through this situation that you thought that this would pan out, but it did not. And that uncertainty is going to always be there in a company such as ours. So, what is your experience in really putting together a business case with so much uncertainty in the pipeline?
Put one other person on the spot here. So the reason I bring this up, right, is I think a lot of the Lilly small molecule continuous investment was made in small volume continuous for traditional small molecules. But as you showed on the slide, there is a very exciting peptide, tirzepatide, being manufactured, maybe similar equipment, maybe not, but with that same fundamental understanding of CM. So there was this transition, right? Any thoughts on how you navigated that?
Yeah, I can speak a little bit to that. I think your point is very well taken, right? In innovator companies, right? The portfolios are diversifying. We're seeing a blending of this traditional small synthetic molecule space and biologic space, right? With all the new types of therapies that are emerging just everywhere, right? But I think at the core, when you think about how you're going to implement, build a CM, organization or infrastructure in a company, especially in a CM&C type of space, right? The thinking, the integration, the cross-functional engagement allows you to kind of build and develop a philosophy of how you're going to tackle other processes, molecule agnostic, right? Whether it's a peptide an oligo, an antibody conjugate of some sort. It's a way of thinking and problem solving and how you quickly triage where can you innovate and implement to make something better or elevate a capability or connect to types of unit operations that if you were just focused strictly in a batch sense, that you would take on differently. So I think that's an area that we've been able to really capture and harness, right? Like we just don't work in SVC of small molecules. We apply what we've learned in this cohesive, innovative mindset to our entire portfolio, in the decision making of how and where we can implement it to do that work is driven by the business, right? We don't as scientists go, oh, hey, let's just go jump into this, sandbox and start working, right? It's intentional, right? It's not random. And I think if you can assemble a team of scientists that are able to do that and kind of understand that the road may be challenging, but the benefit is potentially very high and could create a step change in how you do CM&C, I think you can be successful regardless of the modality, right?
Well, thank you, Todd. Thank you, Matt. I think an incredible round of applause for what he has put together, as well as you. I mean, we got through 2 questions, which I think was an outstanding, but we've learned a lot. But we also know there's a lot more to learn. I think, when you look at the Amgens and the Pfizers, the Lillys, the Mercks, etc., GSK, your major pharmaceutical companies are doing it. There's a lot invested, but at the end, they've come up with better products and better processes. They're doing it faster. So we don't have all of the answers. I think one of the things to the USB, acquiring case studies to kind of further relieve the pain of what's needed to implement these things. So I guess we're on a break. Dennis, do you have anything you need to say, or?
So, well, yes, we're about to go on break, and so I want to thank both Matt and Ernie for taking us through that today. And just to remind you, from a schedule standpoint, this is our only break before the end of the workshop at 1. And so take good use of this particular break. We're going to work straight through the next sessions. So thank you very much.
Seconds. Business.
40 in continuous, and then there's regulatory at 11:35 to 12:30. So I guess we'll see how this next one goes, to see if we actually want to listen through to the end of it. Great. So it's another 10 minutes. Okay.
If we can just return to our seats, we're about to get started in just a few minutes. Thank you.
Oh, there's one.
Well, welcome back. The next session, we're going to have Lawrence DeBelder talk about quality. Although quality in batch processes has been to be proven to be fit for purpose, there is a general belief that continuous manufacturing has the opportunity to improve to reach a higher quality profile with a narrow distribution, higher consistency, and lower risk to failed batches, recalls, and out of stock. The process understanding is better, which also makes it easier to resolve investigation and identify adequate solutions. Because the higher level of automation, there is less risk for human errors, and because there are fewer or no intermediates, also there is a significant risk reduction. So the quality aspects are not a barrier to the adoption of continuous manufacturing, but the lack of understanding how to get to what a control strategy could be, or the hesitance to change by the QA, QC, or regulatory organizations. I will now like to turn it over to our colleague Lawrence for a brief introduction and his input, as then we will enter into the Q&A. Thank you.
Thank you, Ernie. So my name is Lawrence DeBelder. I work for Pharmatech Associates. We are a consulting company that was acquired by USP to help the industry and to overcome the barriers of adoption of continuous manufacturing in reality. And well, we can do that through business strategy support, through helping development of products on continuous manufacturing lines, and helping with the regulatory strategy as well. So I was asked here to give a bit of a reflection on what are the barriers of continuous manufacturing related to quality. Well, I think there are none, because the quality improves when you implement continuous manufacturing in reality. So, but before going into details of that, I want to take a little step back and reflect on how did the world change over the last years. How does that impact the way that we need to implement advanced technologies in the way that we make medicine in reality? So one of the things that changed is the more and more scrutiny on quality of the products. So with the health authorities all over the world, they are focusing more on that the patients get high quality product. And they're getting better and better in zooming in the details of manufacturing processes. Not only with the innovators, not only with the generics, but they are moving further and further along also to nutraceuticals, to OTC, and there's more and more investigations going on, especially after COVID now. If we look at different regions in the world, there's some regions in the world that have quite a lot of issues that were discovered once that the investigations after the COVID period came along or beefed up again. Quite some things were discovered. So more and more scrutiny on quality, and for good reason. Of course, we want to protect the patients everywhere. Now, the supply chain issues that became clear during the COVID period also had results. There's many programs, government programs that try to onshore or nearshore the API production or local manufacturing of medicines as well. That's as well in US as in Europe a hot topic. And besides that, generics market is growing, especially in Japan. Initially many years ago there was nearly no generics in Japan, and now generics are very much growing there. And there's an increasing competition in the world between generics and of generics, but that makes their margin even smaller, with potentially a higher risk for quality if depending on how it's managed. So there's a lot of things going on. There's an evolution in the world, but still, if we look at continuous manufacturing, that is being seen as one of the potential solutions to all of these issues. What products have been approved until now? Not so many. We are at a list of 20 or less in general. You would say that it's surprising, because if that is a solution for quality issues, a solution for cost issues, if it gives so much flexibility in your supply chain, if it helps with resilience, why is the uptake less? And the uptake is not very high at this moment in approved products on the drug product side of things. Drug substance even less. We talked a little bit about the complexity of that, because there's so many different chemical reactions. You have a very diverse set of equipment that you would need to be able to do a continuous manufacturing process end-to-end in drug product manufacturing. Biologics, it's even worse. It's even more complicated, because there you have sterility aspects, you have a living organism very often that plays a role there as well. So that makes everything even more complicated. Maybe even more fear of changing the process, because the process itself is even less understood in some cases. So the amount of approved products is limited at this moment. We see it growing, and we see it coming and evolving very strongly. Our product industry at this moment is very strongly going towards a more widespread adoption of continuous manufacturing. Almost all innovators have drug product continuous manufacturing equipment in-house, and the smaller and mid-sized innovators, they are evolving in that direction. We also see more and more interest of generics. They're still hesitant, because the business case really needs to be there. And as was said before, the business cases that are published, that are clear and positive, are not that numerous at this moment. But going back to quality, why is continuous manufacturing really an enabler for next level quality? To a continuation of the enabling for different types of chemistry that Matthew brought. I think continuous manufacturing can be an enabler for next level quality because of many reasons. One was mentioned already by Eric Jacob yesterday. Blending capabilities in a continuous manner in drug product, they are clearly a lot better. Many studies have been done that there's big advantages there. Also for continuous manufacturing of wet granulation processes, many papers have been published that a narrower particle size distribution can be achieved, and therefore also better control over quality. You also have less startup and shutdowns, because your batch size can be longer, and because of that you can have a more consistent production, and therefore more consistent ways to stick close to the CQAs that you would like to have. You can more easily scale. That was also mentioned by Eric yesterday, especially in the blending area. That's a very important aspect. For me, most important reason is because of the continuous manufacturing processes that you put in place. You have to install PAT or a lot higher resolution sensors, because you have to make sure that you understand your dynamics within your process. And because you have this higher resolution, per definition, you will understand your process better. it is looking like, it is maybe the difference between gazing to the stars with binoculars, as we do today with batch, versus taking a Hubble telescope and looking at the details of the stars itself. And so you can have a lot better understanding of all the dynamics in your process and how everything interacts and all your variations. And in that, with that knowledge, you can steer your process to be right in the middle of the sweet spot as well, and make sure that you stay right there where you want to your process to be to achieve the best CQAs that you have. And also during development, while you are doing your design of experiments, normally you have to make batch after batch after batch with a different set of parameters. With this, you can do changes of parameters on the fly, which gives you the opportunity to do a lot more design of experiments with the same amount of material, which gives you the opportunity to design a much more robust process with the same amount of material in your development. There's also the possibility that because you measure inline, that you can partially reject the portions of your batch that are at less quality or that are out of specification. You reject those, so your overall batch will be having a more accurate set of CQAs and will be more within spec or closer to the sweet spot of your specifications as well. And because you know so much of your process with all that data, you will also be able to resolve investigation afterwards. So your process knowledge is not only better during development, not only better during the production itself, also if you have something that goes wrong, you will be in a lot better place to understand how to solve that and solve it faster as well. So how is that in any way less quality, or how is there in any way a barrier for the quality aspects of continuous manufacturing? Not at all. I think it's only better, there's only improvement if you implement continuous manufacturing. Now what can be a barrier? The barrier can be the knowledge of this. So the knowledge of the understanding of what does it really do? Does it really improve my quality? Does it really improve my process? And how to do it? How would I have to build a control strategy that is all-encompassing for this continuous manufacturing process? For drug product, there's already a lot of papers produced and published related to that. For drug substance, a lot less. For biologics, non-existing. So that is probably one of the biggest barriers for adoption related to quality at this moment, I think. But quality-related aspects is not everything. I want to take again one step back to give a bit of an overview. What I think are the three big buckets of barriers at this moment. One bucket is the financial aspect. It has been discussed already by Matthew for drug substance. We had a bit of a discussion on drug product. Maybe there's a few more additional things we can say about what are the drivers of the business case for drug product continuous manufacturing projects. But in average, what the feedback is, if you hear hesitance of the adoption of continuous manufacturing, is that the business case is not clear. That's mainly the biggest discussion. So it's not clear enough for the C-level of people to accept to invest in that. Why is that? Because the cost is not always clear and the benefits are not always clear. So from the benefit side of things, it's based on assumptions. Those assumptions, to what are they compared? There's not a lot of data available, and these assumptions, well, to what do you compare? To batch is the best way. But if you have a new technology and a new process, how can you just have to guess how it would look like in batch? So the assumptions that you make for your benefits, they are not always very much data-driven and more guesswork than something else, which is sensed, I think, by C-level managers as well. So that's in some cases why they push back. On the cost side, you have the equipment cost, but there's a lot more. And so there's also the cost of transformation of a full organization. There's the cost of licenses of software that you don't know of in the beginning. There's the cost of all different aspects that you might not always think of. And many of those early projects, they the costs could get out of control, because initially they were not thinking of all the costs that are part of this journey. So if costs are not clear, benefits are not clear, the business case is not clear. So over time, I think that will be solved by itself, because more and more of these business cases will be published and discussed. Also, the prices of equipment will drop, which will help these business cases. You see you see that happening in the drug product side of things very strongly. And nowadays, you have an equipment on the market for $4 or $5 million. You can do direct compression. Already, you can do direct compression in a compression room. That's not comparable with the cost of equipment and the impact that it had 5-6 years ago. So your business cases overall become better, your benefits become more clear, and I think that that's only for the better for adoption. And I think that for sure will help the generic industry also to adopt this over time. Second big bucket, I think, is the knowledge gap. And so and that's present everywhere. We discussed yesterday as a feedback of the workshop that the knowledge of health authorities in different areas in the world outside of the ICH group is not always there, and sometimes it takes a lot of time and a lot of effort for companies to convince and to explain and to get to the details with the exotic health authorities. And that might be a big fear for global rollouts, because if you need to do a global launch and you are certain of the FDA and EMA, etc., but you're not certain about the number of countries, what are you going to do? And so you need to make sure that you can launch everywhere. Another knowledge gap is the resources. So you want to attract people that have experience. You want to, if you want to build out continuous manufacturing, you need to have the knowledge in-house, but those resources are not always available and not easy to get. Also, external knowledge at CDMOs or external knowledge with consultants is not so easy to get. So there's a limited amount of consultants to support. There's a limited amount of CDMOs that have either the knowledge, the experience, or the type of equipment that you are working with to support you fully with your journey. And so there can be this lack as well. And then the internal knowledge, how to do things, what to do, it's not always very clear. The pathway, how to get your product filed is not very straightforward. The documents, the guidance documents that are published, they talk about, as we said yesterday, about the how, but not exactly about they talk about the what, but not exactly about the how. And even how to build a control strategy, step by step. There's no document out there at this moment yet that describes that. Luckily, USP is working on a guidance document that is helping in that regard. And also, there was no individual local one-stop shop where you could find all the publications and all the documents and all the guidances that are published related to continuous manufacturing. Also, that USP has solved with the Knowledge Center, where all documents that are public are now in one place, which is free to consult, and that's a collaboration of USP and NIFT. So that's the knowledge space, and then the regulatory space. There's also, I think that's basically driven by fear, and so companies do not really know what the expectations of the health authorities are. They are afraid that the approval timelines will be longer than for batch, which yesterday was clear that on an average, all the filings that have been done until now were on an average 3 months or more faster approved than batch processes. So based on fear mainly, and then but I think the biggest fear is the not understanding of the requirements of the different health authorities outside of the ICH Q13 or the ICH group. And what would be a good solution there is if there would be one party that would map all the differences, specifically all the discussion points in continuous manufacturing discussions. That would be great if we would be able to evolve to that. But I think that's the overall barriers in general. Now, if we zoom into the quality barriers, there is let's go through them, and I would like to have your opinion as well on this. So I think number one is the understanding of what is really needed to achieve the quality level, what is really needed to build a control strategy. How far do you need to go? What data do you need to file related to that? What data do you need to collect? When is it enough? Basically, that's the biggest question, I think, related to quality, which overlaps a little bit with the regulatory aspects. And let's instead of going through that list completely, I would like to have maybe the feedback of one of you. What do you think are the biggest barriers quality related? When you go through this list, and we start with number one, the understanding what is needed. Do you all in the audience have a good understanding of what is needed to do a continuous manufacturing process, of what you need to file and how far do you need to go? Or do you have the feeling, well, it's not at all clear, it holds us back? And maybe that's one of the drivers. Anybody from the audience would like to comment on that? So so let me vote. Let us do a vote then. Who has the feeling that they have a very good view on what is needed to achieve the quality in a continuous manufacturing process? Put up your arm. It's a very small amount. And who has the feeling that it's not clear at all? And actually, there's not enough publications. There's not enough knowledge out there at this moment to know exactly what you have to do. Could you lift your hand? Okay. So that brings us to the second bullet: fear. Fear of the unknown, and I think it drives a lower level of adoption, because that fear of the unknown is very present within the organizations, and that starts with the QA organizations. I think let us keep doing what we're doing. We're certain of that. Why change? The quality of batches is good enough. And I have to emphasize, it's not that the quality of batches is bad. It's not because we are gazing at the stars with the binoculars that we need to switch to a Hubble telescope. But in some cases, it is beneficial for the patients, and in some cases, it might be needed. So if we start from risk assessment from the product, risk assessment from the process, and we start from what are the risks for the patient, and we trickle back, it might be possible that continuous manufacturing gives you a tool that we did not have in the past to achieve the level of quality that is needed for the risk level that the patient is working with. So back to the quality organizations in the companies, fear of change, fear of not understanding, fear of maybe not improving, and fear of all the implications that it has. So that would be the next question. Do you think that the quality organizations, the regulatory organizations in the companies, do they have fear of change, and is the hesitance for a strong part coming from them? Then the next question would be: is it once that you have implemented a quality discussion could be maybe the fit for continuous manufacturing lines. Maybe you don't find exactly the products that fit on the line. One of the issues could be the bad flowing products. So if you have API that is not well flowing, doesn't really fit in a line, or it's a lot more difficult if you have different APIs in one product where you have a fingerprint that is overlapping. If you have a very low concentration of API where your PAT cannot measure it, so these could be difficulties as well. And you need to align your R&D and your commercial organization. Your R&D organization prefers to have flexible equipment. Your commercial organization prefers to have efficient equipment, and that's not always overlapping, or that's not always the same. So that alignment might be difficult as well. And then the experienced resources, finding them, and
everything that is evolving all the time might be a perceived risk as well. So I think those are, in general, quality barriers that can be perceived and that could lower the overall adoption of continuous manufacturing. So that's my opinion, that's my view at things. So I think we can now switch to the questions of the audience.
Thank you, Lance. I think if you advance your slides, we'll get to the Slido questions. Go to the next one. Okay. So, level of training and capability, especially for those that are comfortable with status quo. You know, any thoughts on that? And what we have about, what Lawrence and I have talked about, that we have about 11 questions to get through. It's highly unlikely. But do you have a good overview of what are the differences between batch and continuous manufacturing, and what benefits continuous manufacturing can bring you?
Well, that's a very broad question, and that will take a few minutes to answer. So, first of all, the biggest difference between continuous manufacturing and batch manufacturing for drug product, in any case, is that you have a constant stream of products. So you have process dynamics for continuous processing versus in batch. You have everything in one go. You mix, and then you sample, and you measure after each step. So that's a big difference. Because you have a dynamic stream, you also have the risk that in those dynamics, an excursion comes in and propagates all the way through to the end of your process. So therefore, you need to measure at a high frequency to capture those variances. So the measuring that you do, or the sampling that you do, is because you need to measure the changes in time. You have to assume homogeneity in time versus homogeneity in space, where you in a batch process measure in different locations in a vessel to assure that it's completely homogeneous. So I think that's the overall biggest difference quality-wise. And then what are then the biggest benefits for continuous manufacturing versus batch? I think the quality ones I already discussed, but there's many more, of course. You can run at a lower cost. You have flexible batch sizes, which improves your resilience, your flexibility in your supply chain. So you can do faster development. Your tech transfer is 0 to nothing if you have the same equipment in batch and in development and in commercial manufacturing. So that brings you also to a better speed to market, which is the biggest benefit driver for many companies. So that in a nutshell.
From the audience perspective, any comments, any thoughts?
I actually just wanted to come back to that question up here that's voted. So coming back to training and capability, I'm wondering if this is related to quality systems, or there are other functions that are maybe slower to get away from the status quo. That is right, because I guess my experience has been those in the technical functions are generally eager to engage, try new chemistry as an example. But are there other functions that you've experienced or others experienced that are maybe slower to get on board?
Well, there's a big difference between an equipment in the drug product side in any case. So there's a big difference between the capabilities of an operator in the batch world versus in the continuous world. Because in the batch world, you have, for instance, an operator of a compression machine that knows his machine inside out, that can hear that something is wrong and knows exactly what knobs to turn to get it perfect again. In a continuous line, you typically have operators maybe 2 operators that run a complete line. So they need to have the knowledge of all the different unit operations, Plus, on top of that, the understanding of what the PAT does and what the output of the PAT can mean. So the PAT aspect is a new aspect there, and that raises the bar of the knowledge of the operators. That's one thing. And then a second thing is all the modeling aspects are relatively new as well. So the building of the models, especially on the R&D side of things, that can be relatively new for an organization as well.
Just to hit on this in a talk yesterday, but this is a really important thing here, which is that you said that there are dynamics in continuous manufacturing. I think what's different about continuous is that when you're running at a steady state, there's a lack of dynamics, and it's easy to maintain. In batch, like spray granulator, the entire phase is dynamic. If you lose power in the middle of a wet granulation, the whole batch goes down, you have a problem. If you lose spray rate, you're outside your space. So this is actually an advantage, and the idea that that continuous needs more control in place because it has dynamics is actually the opposite. It's that continuous is easier to control because you're looking at set points and they're just in one place, whereas everything else that has a full batch process, you're running through a dynamic trajectory and trying to match that trajectory, which is much more challenging. So I think it's I'm sensitive to it because it's one of these things where it's a technical advantage and it gets framed as a weakness.
Okay, moving to the next question. The response at the top, ignorance within companies about what pathways to continuous manufacturing really are, need for lower level guidance within the industry. And I'm going to try and tie it into the second question for you, Lawrence. Do you have a good understanding of what is required in this continuous manufacturing filing? Do you see challenges to meet those requirements?
Well, the biggest challenge to meet the requirements of the health authorities in a filing is lack of knowledge. So you need to very well understand what you're doing. So all the statements that you make, you need to back up with data, with strong rationale, scientifically based rationale. And one of the important fields there is statistical knowledge. So because you have to, you need to measure at a high frequency in a dynamic environment, you need to understand what frequency that needs to be, and therefore you need to back that up with statistical information to show that the frequency that you have chosen will capture all the potential excursions that might occur. And also a good understanding of all the potential risks that there are. So all the types of variation that can occur. You need to understand your materials very well. So all the material characteristics variation you need to measure very well up front. So a very strong knowledge early on is an impediment for continuous manufacturing filing as well.
From the audience, any thoughts on what Lawrence has said?
Yeah, so I think the same comment, which is that because we have 50 years of doing this and we have methods that have been specified that are maybe not statistically significant, we have pathways that have been accepted for batch that are not as intensive as what we have for continuous. But at the same time, material properties are just as important, if not more important, for batch. Like I said, what I see in continuous is that we can detect it, but we actually can deal with a wider variation and keep the product quality on there. So it's a thing that you should study your material properties, whether you're batch or continuous. You have more tools in continuous to do this. We lack tools to study material properties at the commercial scale because we can't run 10 experiments at 300 or 500 kilograms of scale. So again, just a phrasing there that the advantage is that you can do that in continuous, where we can't do that at batch at commercial scales in an economically viable way.
Yeah, fully agree, but it's still the case that the health authorities require it at this moment.
I think, when you look at the health authorities and what they say, nowadays the newer guidances are about risk assessment and about going forward and, and doing those things. They're becoming very similar, but you still do have health authorities saying you can follow the guidances of the past. And that again gets into the fact that this is where the unlevel playing field is that you can go through this thing and kind of hope that you get what you want out of your sampling and get a product online. In a batch paradigm, that option doesn't exist in continuous times.
Excuse me, can virtual members participate too, or are we allowed to ask questions?
Yes, please ask.
Okay, this is Fernando Murcio from Rutgers University. I've been trying to participate for a while. So I think that we are supposed to be having a discussion across all the applications of continuous manufacturing, not just solid dose. But when it comes to solid dose, I would say branded pharma has gathered more than enough experience on how to satisfy most of the main regulatory authorities. The question comes up when you go to other places like maybe Brazil or Turkey, where indeed the regulators there haven't had enough experience. But I would say US, Europe, Japan are pretty well, the regulators are pretty well on board, and I think the largest pharma companies are also pretty clear. The question becomes whether the new companies, the companies that are beginning to do this now, whether they have the in-house knowledge or can they access it easily. And one problem is that the knowledge is not easily available on what to do and how to do it and how to meet the standards. So that's for solid dose. I think that we're going to see in the next few years an evolution towards automated real-time quality control systems that are going to make the job easier for everybody. I think the statistical knowledge is relatively easy to gain once you have people, looking into it. Now, in API, I think the situation is quite different because, and I'm going by, for example, we have several projects funded by FDA to create digital twins for API, and we discovered, much to our surprise, that most of the companies doing continuous flow are not actually developing kinetic models of the chemical reaction, where they actually do gain the ability to express the rate of reaction as a function of instantaneous reactant concentration or solvent or temperature, etc. It tends to be run more as a black box, and that limits the ability to both control quality and scale up, and it potentially misses one of the biggest potential advantages of continuous flow chemistry, which I have not really heard too many people talk about, which is the opportunity to minimize the production of impurities. One of the advantages of continuous plug flow reactors is that they achieve much faster mixing than anything you do in batch. And we know as chemical engineers that if you run the reactions very fast, if you have fast reactions and you achieve very fast mixing, your yield should improve, but more importantly, your production of byproducts should decrease. And this is a unique advantage of continuous plug flow reactors relative to batch stir tanks, but it requires you understand the kinetic model to be able to actually optimize the conditions at which you're running. And the industry has been running continuous flow reactors for many years, but has not really developed the capability to create these kinetic models. Yeah, and in bio, I would think, just to cover the gambit, the main issue there is that we don't have the rapid sensors that allow us to monitor what's going on in the bioreaction in real time. I mean, we can monitor dissolved oxygen, we can monitor, glucose, temperature, and pH, and things like that. But it's very hard to assay the main product and the byproducts and things like that. So we don't even have visibility over the instantaneous course of the bioreaction, which makes it even harder to control, because if your reaction has a characteristic time of, let's say, 2 hours, but your assay takes 6 or 7 hours, you don't have an assay that can be used to surveil or monitor the process. So that's where I would put my relative evaluation across the 3 main categories of continuous manufacturing.
Thank you, Fernando. Lawrence?
Thanks a lot, Fernando. Yes, thank you for this addition. I until now only spoke about the drug product side of things, but in drug substance, that's very clear. So, any other reaction from the audience on where do you think that that quality might be a barrier for the adoption of continuous manufacturing?
I think the feedback from the online contributors very, very well received. Just maybe one or two comments that maybe turn into a question or two, and I'm curious others' thoughts here. So one is, I think it's important that we acknowledge that quality of batch manufacturing is held to an incredibly high standard. And so I don't think operating in CM is inherently higher quality. Another attribute associated with CM is inherently safe. And I think that's also not true, right? There's other safety considerations that need to be taken into account. And so we have decades and decades, if not more, of quality batch manufacturing, so that's good. The quality considerations for CM, I think, are just different, and our ability to probe, build models, add additional PAT is helping us understand the processes better and impart additional quality metrics or considerations, perhaps. I guess the question I have, and this is aligned with what Eric has brought up earlier, it's just because we can, do we have to? So the example I shared is a DS manufacturing process relying on parametric control only. The very first question we get from the agency is, where's all the sensors? Where's all the PAT? The next is, we did a lot of experimental characterization of that process. Very traditional approaches, one factor at a time, assessment of these different parameters, and built an experimental knowledge state to support our control strategy. The second question from the agency was, where are the process models? And so, again, because we can, do we have to? I think that's an area of unknown for me. Very much open-ended statement, but I'm curious, thoughts on that?
Yeah, thanks, Matt. And I just want to add to that comment on quality. So while we say CM has better quality for a drug substance, right, any chemical reaction, it is true in some cases, but we've also come across cases where, because the mixing is better, the kinetics are faster. The kinetics of parallel reactions are also faster. So we are seeing, if you look at the HPLC, like the purity profile, in a few cases we are seeing many more small impurities that are showing up in the flow process as compared to the batch. And in one of the cases we worked on, which is a butyllithium reaction at low temperatures, we are not quite able to match the purity that we're getting in the batch process, which is really, I think it's perfected. And while of course this works in flow, the impurities get washed away, so it works. But we are not able to quite match the purity profile that we're getting in batch. And we've discussed this with many experienced chemists and tried different combinations, but couldn't quite crack this problem yet. So that's just a comment on the quality. And so I'm just saying it's not true for every case that the quality is better in flow, and that's been our experience. Also, the second comment on kinetics is, while we have done kinetics, what I alluded to in my presentation also yesterday is, it's very, very hard to get a kinetic model that's sensitive enough to these very low levels of impurities or starting materials. Like, we wanted the kinetic model to be able to pick up the consumption of the starting material down to below 0.1%. And to have a kinetic model that's going to come to that level of precision and accuracy is really challenging. So, how do we build such precise kinetic models? Because if you want to use the model then for anything, any predictive capability, it needs to be able to, predict below the spec that we're putting. So how we build such, kinetic models is also a challenge that we face.
So let me respond to that. I'm sure that you're right that there are cases like that, but I also think that there are many other cases where there could be significant benefits from having a good kinetic model that can be used to optimize the reactor to predict what are the best conditions to operate. So just because there might be some things that are difficult to do shouldn't prevent us from taking the next big step towards developing a full model of the process and really using all of the available tools to achieve an optimum result. It will be a case by case, right?
Yeah, no, absolutely. So I'm not saying we shouldn't do it. I'm just saying, So we did develop a model, by the way. We did develop a kinetic model with JPROMs with, residence time distribution built in, and we did all of that. So it's not that we didn't do it, and we spent months getting, data to get to that precision. I'm just posing that it's more challenging than it appears to say a kinetic model. Whereas a kinetic model for a typical batch, just optimization, on a broad level is much easier.
And maybe we're talking about different things. I mean, you mentioned GPROMs and RTDs. That would be a macroscopic dynamic model of the process. What I'm talking about is using stop flow chemistry and high sensitivity instrumentation to really understand the reaction rate. You don't use GPROMs for that. You use basically genetic experiments to try to elucidate the equations that describe the rate at which the reaction proceeds. You identify the elementary steps, you identify relative reaction order of all of the reactants and intermediates. That's what I mean by kinetic model.
No, that's exactly what I was referring to when I said kinetic model. I said we then built it along with RTD and other things into JPROMs, but the kinetic is exactly what you mentioned.
Okay, just to make sure we're talking about the same thing.
Yeah, same thing.
Okay.
Let's move on to the next question. Lawrence, is continuous manufacturing embraced by your QA department as an important improvement towards the future?
So I think that's a very important question. So and I wonder if that is everywhere the same. So it's clear that higher process knowledge can be achieved with continuous manufacturing, and potentially better quality can be achieved with continuous manufacturing. And the question is if the QA organizations can see that as well and are willing to go through that transformation of getting all the learning in and having to look at batch records and process data in a completely different way, because there will be a lot more data, there will be different types of data that needs to be assessed. So there's a big learning curve for QA organizations as well. And I feel here and there quite some hesitance from QA organizations to go in that direction. But to which extent that will evolve, that's one of the questions I have for the audience, actually. It's curious to understand what that is in different companies.
So I don't know that I felt hesitancy from QA organizations. I think they're usually pretty on board with the positive control that you have over your continuous implementations. What I felt is QA organizations are responsible for a lot of on-the-floor support with deviations and getting batches released, and that they don't necessarily have the bandwidth to come in and do a transformative project. So for me, it's not about attitude, it's about the fact that a QA organization is vital to the day-to-day operation of a plant, and that they have a 45-hour day job that involves getting batches cleared and released. And so there needs to be some sort of support from leadership to get people onboarded, trained, and take a loss of efficiency in them doing their day job.
I do see in some business cases that there is a saving in the business case related to the amount of QA support that will be needed in the future. So although that it becomes more complicated or different to look at the data for these manufacturing processes, there will, if you are able to make a lot longer batches, you have less batches. If you can connect all the unit operations to each other, you don't have intermediates anymore that needs to be released. So there is, in reality, less batch records to be reviewed. There's less intermediates to be released. But if you integrate also real-time release, there is overall a lower release effort to be done. And because of the higher process knowledge, investigations are also easier solved. So because there's more data available, and it's easier to get to a conclusion there. So the question is then, after that ramp up of the initial learning curve to understand better how to do your release and your quality assessments, will there be a lower effort overall for the QA organization? In a number of the business cases that I've seen, that was the assumption.
Can you hear me? Okay, yeah. Hey, Lawrence, I also wanted to mention that I think a lot of conversation between batch and continuous manufacturing here, I feel like has been focused on comparing the two for the same drug product that has a foreseeable quantity that needs to go to the market. There are a lot of cases that continuous manufacturing might have inherent advantages to advance to batch. There are also cases that sometimes batch manufacturing might make more sense. So to I guess follow up on one of the panelists, do we have to do it every time? No, I guess it depends on the case. There are, for example, cases for the drug products that the demand in the market fluctuates, and it is very hard to scale up batch processes. But for continuous processes, you don't have a batch size, you just have a throughput and a runtime. So if you have, if the demand increase, a drug goes into shortage, for example, you can you can increase your output, either run it at a faster rate or run it just for longer time. As simple as that. The other thing I wanted to also mention is that typically, this is not always the case, but typically continuous manufacturing requires like smaller footprint. So if you consider like supply chain issues, does it make sense to batch produce your drug product in one location and then have it transferred to the target market? The alternative would be to maybe have a smaller market, for example, produce in India for a drug product that is intended for the US, but if you have a smaller footprint in the US closer to your market, that eliminates some of the supply chain issues, which could be helpful in terms of building a business case. So I guess it depends on the case. There are also, honestly, some cases where the drug product is very specialized, the quantity is so low that maybe continuous manufacturing might not be the best option. You might want to, continue with your batch product. If the quantity is low and I guess the demand doesn't change from the patient side, then maybe that might be a case where you stick with the batch. So I guess it really depends on the market, but there are some inherent advantages to continuous manufacturing. Like I said, you can have it closer to the patient, you can have it closer to the footprint, a smaller footprint, and you can also have eliminate some of the supply chain issues, and you can have modular production as opposed to batch.
Thank you for that feedback. Yeah, very valid remark.
Lawrence, we only have a couple of minutes left. A quick thought on technology and the introduction of new technology. Would it be feasible to make a joint project for all the organizations that will be impacted by the implementation of new technology?
A joint project together with different...
Yeah, like process development, manufacturing, and QC, etc.
Yeah, I think that's often the best way forward for larger organizations. That there's one joint project that is defined that is happening in R&D and in commercial and with QA, because it's often a transformation of the complete way of producing. That's one thing. But also because there's a big game in the middle between R&D and commercial in the tech transfer activities and the speed to market that makes it very interesting for R&D and commercial to work together. And that's often a big issue because they have different objectives. R&D wants to have flexibility, they want all the tools and all the whistles and bells to be able to make all the different products that they can think of. While commercial, they want lean machines and they want to have a throughput and production and everything as efficient as possible. Not too many PAT tools in there, no complexity. That's very different objectives. But at the end, they will need to be able to have a joint project, because the biggest gains are coming from copy-paste equipment in R&D and in commercial manufacturing. So if they see that as a joint investment where they reap jointly the benefits and distribute then the benefits together, I think that's the best way forward. So for some companies, like for Vertex, that's relatively easy, because that's more one, that's a smaller organization, or they are closer together. For very large organizations, maybe like J&J, that was more difficult, because that's very separate organizations, R&D and commercial. So to have them work together very closely as one joint project, that was more complicated.
Any last comments?
So I think in general, continuous manufacturing is there to stay, especially in drug product, drug substance. We see it emerge more and more, and there's a number of companies that are picking up and go all the way. Other companies that are doing parts of the process in continuous, maybe will never expand to end-to-end. And then in biologics, it's coming up in the future as well. There's a lot of assessments going on. There's a few front runners. So overall, I think the industry is evolving with the adoption of continuous manufacturing. There's a lot of work that needs to be done to lower those barriers on the return on investment side, on the knowledge side, and on the harmonization globally of the health authority requirement side. But I think we are going in a good direction. And I hope that especially by collaboration of all companies and academics and health authorities together, we will be able to speed up that adoption.
Thank you, Lawrence.
Thank you.
Next, we're going to get into the regulatory segment, and so Riley is here as a follow-up and kind of to shed more light on what Adam was discussing yesterday. It was great that Adam addressed some of the questions that I'm sure many of you had, and I'll just focus on the myths that Adam talked to us. The FDA is not mandating continuous manufacturing. Continuous manufacturing is not just for innovative companies. Generic drug companies are not required to implement continuous manufacturing. CM applications have not taken longer to review and approve. So that kind of gave us gave us more information and understanding, but there's a lot more questions that we all have. And I will now turn it over to Riley.
Thanks, Riley. All right, good afternoon, everyone, or good morning, still, I guess. So I'm Riley Myers. I am Chief of FDA's Advanced Pharmaceutical Manufacturing Lab. I'll talk more about that in just a second. My prior role, just as a disclaimer, I guess, my prior role at FDA was an assessor of biotechnology products. So any comments I make is really through that lens, which, everyone here knows can be a lot different than small molecules. I'm also a member of the Emerging Technology Team in CEDAR and the FRAME Initiative, which I'm sure we'll talk about at some point. So FDA is constructing a new advanced manufacturing laboratory that's about 10 minutes away from its main campus in White Oak. The laboratory is still under construction, but there were space limitations at White Oak, and then there were other considerations that triggered the need to build this facility or this laboratory. And so the idea is to simulate a state-of-the-art pilot manufacturing plant in order to perform research on pilot scale equipment. And then another vision for this laboratory, which I'll talk about in a second, is to train assessment staff. So first, Adam talked a lot about this yesterday, but I'm going to just quickly spend 4 slides talking about what FDA has done so far to facilitate the adoption, really, of advanced manufacturing technologies in general, but the focus here will be CM. So the first two, which we'll talk about more later, so I'll just briefly mention them here, are CEDAR's Emerging Technology Program, and we partner routinely with CEPR's Advanced Technologies Program. They are also dealing with CM processes for their products. One of the challenges with adoption of any advanced manufacturing technologies has been mentioned here. It was mentioned yesterday and several times today is international harmonization. And so FDA routinely participates in that process through ICH, and documents that are applicable to continuous manufacturing are Q13. Q5A also includes information on bioclearance for CM processes for biotechnology products. Q14 includes information related to multivariate models for analytical procedures and real-time release testing, which can also be applicable to CM. Then ICH Q12 also includes information on lifecycle maintenance of process models. FDA over the last 10, 15 years has published numerous guidances on technologies, advanced technologies that can be used with CM or CM itself. These include PAT, process models, and real-time release testing. FDA research also contributes significantly to mitigating adoption or mitigating barriers and facilitating adoption of CM and other advanced technologies. And so I think Adam showed the slide yesterday, which is why I chose to omit it, but projects, internal and external funded projects at FDA generate more than 78 internal reports and publications. On top of that, FDA routinely reviews the impact of intramural and extramural research on the adoption of advanced technologies, and we've published at least 9 in the last 7 years or so on average more than once per year. And that's not just on FDA's in-house research, it's also on external publications. FDA research contributes significantly to workforce development. So training and assessment staff already occurs, but part of this new vision for this new laboratory is to have a sustained training program so that all the general pool product quality staff, product quality assessment staff can be exposed to these technologies and not just the subset that are part of the Emerging Technologies Program. Although knowledge in the Emerging Technologies Program is routinely shared with the general pool of assessment staff. Research also contributes to policy development, including ICH Q13 and other things. And then also is generating responses to 15 or so ETP projects. Looking forward to the future and beyond in continuous manufacturing is the FRAME initiative at FDA. Adam touched on this, or talked more about this yesterday, so I won't go into much detail. But part of the challenges, which is what we've heard yesterday and today, is merging of different advanced technologies with CM, so implementing PAT and artificial intelligence and process models. And one of the primary focuses of FRAME is artificial intelligence, and there's various concepts in those 4 technologies that we're focused on that could apply to CM. For example, Matt mentioned earlier about how to standardize equipment to reduce quality risk. One of the areas that FRAME focuses on is a technology called distributed manufacturing. And a foundation principle of that type of technology is standardization to reduce quality risk. So even though FDA has participated heavily in this area, the nature of innovation is that there's always going to be challenges that industry and FDA need to face. It's just the nature of evolving technology and science. And so here's shown on this slide are some challenges, regulatory challenges that FDA has already identified. This slide was originally drafted for advanced manufacturing technologies in general, but these certainly apply to continuous manufacturing. One of the things is how to ensure that these technologies operate within the existing regulatory framework. So for continuous manufacturing, as again has been mentioned, is how to ensure that material versions and quality investigations comply with CQB requirements. Process validation, of course, is a significant challenge for many CM processes. We also have post-approval issues, such as inspecting new types of technologies. For example, the Office of Regulatory Affairs may not have extensive experience with continuous manufacturing processes and changes to the quality system that may be needed to accommodate those. Of course, lifecycle management is always a challenge with any application and with any product, but particularly with new technologies, especially across multiple jurisdictions. And of course, I mentioned earlier that the FDA continues to recognize the desire for international harmonization among regulatory bodies. Adam showed the slide yesterday, so I'm not going to go into detail. Just the additional point that I want to make is that FDA captures feedback from stakeholders and takes specific actions to mitigate barriers in response to that feedback. And this has led, the first approval of the CM process occurred in 2015, and we've accepted 50 proposals into the Emerging Technology Program since that time and approved 13 additional submissions. So there are no regulatory barriers to adoption of continuous manufacturing, only potentially perceived ones. And so I'll stop there and we can sure.
We look at the top vote getters. I think difficulty in of changing existing filings. I think as we go through the question, maybe we can, we can tie it in. There's guidance for batch, and as continuous manufacturing continues to evolve, where is the FDA today in its support for continuous manufacturing, specifically in small molecules?
Well, I think just the FDA support is strong, as evidenced by the fact that we participate in not only producing our own internal guidance, but also in ICH. And even in the summer, we participated in, I think, 4, 5, 6 different meetings and held our own meeting on gathering feedback and providing feedback on facilitating adoption of these technologies. So FDA is very supportive in biotech and small molecules adoption of CM. But as Adam mentioned, it's not an expectation or requirement. We see the advantages of it, and if that fits a particular product and business model, then we are very supportive of developing those types of processes and technologies.
Does the audience have any specific questions? And that also applies to those of you online. Any questions?
Hi. So question about, you talked about how much research the FDA has done, and I think if you've heard from a few of us over the last 2 days, the FDA has done a lot, and it's sort of easy to work with them. I'm wondering if there's anything that the FDA is already doing or could do to share some of that research with other regulators, because a lot of our problems are kind of getting to those less advanced regulators.
Yeah, we routinely publish our research, and that would be the primary format by which we share it with everyone, not just other regulators, industry, and academia.
Any other questions? Oh, okay. Amin?
Yeah. Yes, I have one question. Do you think that we need to interact with the agency early in the development process, or can we, example, just
I don't have it. We found
Oh,
over
my
here.
goodness.
Laura, you should probably leave the door open. Can you leave that door open?
Oh,
Just
yeah.
because it's very warm.
Oh, yeah, I don't see why.
Okay.
That's fine. Yeah, just, we got
Yeah, is Jane coming here or is she?
Actually, she must be on.
There's rooms in there. How's he doing? Somebody left her on. Already major neuroscience. Major neuroscience. I have daily contacts, but I such a lazy butt. Lauren, Jane's on there, Carrie's on there.
Yes, we're gonna, I'm gonna skip back in a couple minutes.
Okay, I didn't see her in there.
We got a couple minutes. Take breaks.
I was like, that's like grandparents. Yeah. I just got her vaccines this morning. But it's like, just with these baby too. Yeah. I know, but it's still better than trying to, that's what I'm saying. It's what I'm saying. It's a shame. Yeah, I think this is Lauren's next year. This is her third. I thought we were engaged.
We're just hanging on a couple minutes for one committee member to work on.
Hey, Essa, can I ask you a question? We have all these sevens we have to screen, just regular MCF sevens, but like toxin resistance, OTC resistant. If, because I would be really.
Folks online, can you hear us?
Can I just ask a little bit of your time?
Yep, you sound good, Lauren. Awesome, thanks, Megan.
I've saw that in y'all and how to run those, and it's the trash one. They just have to do regular differentiating.
All right, folks. Welcome to Sheikah's Defense. I have, before I let her go, and I know we're all excited. There were just things I wanted to say, and I actually found this really hard to compose because there was so much that I wanted to mention about Yoshika and all that she has done and the kind of person she is. And I don't think I can actually put all that into words today, but I'm going to try to hit some highlights of what Yoshika has accomplished here in her 5 years here. So it's my absolute pleasure to introduce Yoshika Kamte today for her PhD defense. Yoshika came to us from the C. Shah College of Pharmacy in Mumbai, India, where she graduated in 2017. And while she was there, she actually did a lot of sort of undergraduate research into natural products, looking at mechanisms of action, stability of different compounds, and that brought her to us, which we are so grateful for. And I will mention that while she was there, she won the Best Undergraduate Student Award the year she graduated, which also just describes Yoshika to a T. But she came to us in 2017, and really she's been a force to be reckoned with since she came here. And I'm just going to hit on just a few of her accomplishments that I think are worth noting. First of all, what she's going to talk about today is a model that she developed in her time in the lab, and I'm going to let you speak into that, and she'll tell you about it. But this is truly something that she took from the ground up and developed herself, and it's really going to shape, I think, the future of the lab and what we do next with what she's done. And I also just want to say from the outset that this is really very much Yoshika's thesis. And what I mean by that is she's always had and demonstrated such an incredible sense of ownership over this project. And the ideas and the development and the work is so truly her own and something that she has just taken the reins on and done. And I almost feel like, you know, you kind of came to the lab in 2017, kind of said, we'll work on this project, and then she just kind of went and did it. And you know, she so truly took control of it and has done such a masterful job on this work. That work's led to 5 published manuscripts, 2 as first author, and a 3rd that is currently in the works. And as she has done so, as a lot of you know, she's essentially been running a flow cytometry core basically on the side, helping numerous other students at Duquesne, helping train them, helping them design experiments, which has also led to other publications for her. During all of this, she's also tutored literally hundreds of PharmD students in physiology, and she also earned an advanced teaching certificate from our Center for Teaching Excellence here. And for those of you that have done it, it is an enormous amount of work to earn that certificate. It's this massive portfolio of different things that you've designed and teaching tools. And she was also awarded the Graduate Student Award for Excellence in Teaching in 2022, which is the highest award that this university can really bestow upon a TA here. So there's numerous other awards in this list and recognitions that we could go into, but I think it's better that she gets to tell you herself. And I will just say, without getting too far into it, that this has truly been one of the greatest privileges of my career to be your advisor. And I am incredibly proud of you and your work, and I'm so excited for you to share it here today. So Yoshika, please take it away.
Thank you, Lauren, for that extremely kind introduction. And good afternoon to everybody. Thank you for attending my defense, either in person or online on Zoom. It really means a lot to me. So today I'm going to talk about how viral infections in the brain can affect development, particularly in children. So several viruses can enter the central nervous system and lead to a variety of neurological outcomes, ranging from epilepsy, encephalitis, like you would see with measles virus, developmental deficits like with Zika, or even behavioral problems and psychiatric problems. And the outcomes of all of these infections depend upon a variety of factors. One of them is the age of the host. For all the viruses that I have listed here, in green, generally, the younger hosts have worse consequences as compared to the older hosts. And when we look at this, all of these outcomes have been recapitulated in several animal models of CNS viral infections as well, where newborn animals succumb to the infection, but adult mice survive the infection. For instance, here with measles virus, neonatal animals infected shows the 0% survival, whereas adults show 100% survival. And this is true for most of the viruses that I have listed here below. When we speak about children in particular, what has been seen is that many viruses that enter the CNS of a child not only lead to immediate effects of an infection, like for example fever, headache, encephalitis, but even after the child has recovered, sometimes they can go on to have long-term effects like memory impairment, motor deficits, cognitive impairment. So a big question in the field is that why does an infection that happen in childhood lead to long-lasting consequences? One of the things that we can think about is perhaps the childhood period is a period of development, where not only your brain but your immune system as well is developing. And both of these systems are needed to fight off brain infections. Part of the problem that the field faces is exactly pinpointing what part of the changing brain or the immune system can lead to these long-term effects. So the goal today is essentially to understand this pathology better with the hope of developing therapeutics to ameliorate long-term consequences. When we talk about the brain in particular, what has been seen is several developmental processes such as myelination, synaptogenesis, synaptic pruning, takes place mainly in the childhood or the juvenile period of life. And this is true not only for humans but also for mouse, where the postnatal period is really crucial for brain development. So we can use mouse to model developmental disorders as the comparisons have been done pretty thoroughly. Now if you have an infection, infecting the brain, the virus is coming in and all of these developmental processes are taking place, one or many of these processes can get altered, leading to long-lasting effects. Along with this, even individual cells of the brain are changing as we grow older, and the cells are essentially maturing with age. One of the cell types that drastically decreases with age are neural stem cells or NSCs. And this stem cell pool declines as we grow older and becomes restricted to specific areas of the brain. So what are stem cells and why are we interested in them? Neural stem cells are the multipotent progenitor cells of the brain, and they have two essential functions: to undergo proliferation to form more stem cells, and to undergo differentiation to form a stem cell and to give rise to a neuron, an astrocyte, or an oligodendrocyte. So as you can see, stem cells really are the ones that build other cells of the brain, and they build the brain in the process. They also maintain this function throughout the lifetime of the host. So now we're in the period of development, which is the juvenile age or the pediatric age. If these cells are contributing to that process, it becomes important for us to know how would they respond when there is an infection or an inflammation in the brain. Now several viruses have been shown to affect stem cell population, either by affecting the stem cell pool, that is by decreasing these cells, or altering stem cell differentiation as shown here. And for all of these viruses, we can think about two possible ways by which they might be altering NSC activity. The first one is direct infection, where stem cells can directly infect, where viruses can directly infect stem cells, and this leads to an alteration in NSC activity. The other is a more indirect way, where stem cells or viruses can infect other cells of the brain like the neurons, and this can lead to activation of innate immune cells like the microglia of the brain, and finally infiltration of the peripheral immune cells into the brain. And the goal here is to get rid of the virus and clear it out. But while doing so, the inflammatory milieu in the brain can also affect NSCs. And my project really looked at this indirect effect of the immune response on stem cells. And I specifically asked, how would these NSCs respond when there is an infection at the juvenile age, and how does that immune system alter NSC activity? So in order to do this, our lab uses a measles virus CNS mouse model. Now measles is a human virus, so humans are the only host. It cannot infect mice directly. So we have these genetically modified animals where one of the three human measles virus receptors, which is CD46, is under the control of a neuron-specific enolase promoter NSC. And this allows the receptor CD46 to be expressed on mature neurons in our model. So the neurons are infected, but the stem cells are not infected. And what this allows me is to look at the indirect effect of the immune response on the stem cells without them being directly infected. The images here are IHC images of a neonatal mouse brain that has been infected with measles. Panel A are measles virus positive cells in red, panel B is neurons in green, and panel C is an overlay image showing that the neurons are infected. Similarly, panel D is measles virus positive cells in red, panel E is now stem cells in green, and panel F is an overlay image showing that the stem cells are not infected in our model. Another advantage that we have with this model is that we do see age dependency in the outcomes of an infection, like we would expect with a measles infection in humans, where children are more susceptible and more prone to measles as compared to adults. And apart from the differences in survival, where we see the neonatal mice die but the adults survive, there are differences in the aspects of the immune response as well. So for instance, in the neonatal mice, there are lower CD4 T cells, whereas in adults, there are higher CD4 T cells. The neonatal animals also have a lot of virus, whereas the adults have lesser virus in comparison. Neonates also have neurological disease, which we have not seen in the adults before. So my question was really to look at the mid-age or the mid-ages, which is the pediatric or the juvenile ages, and to ask how these animals respond to the infection. So I started infecting juvenile mice, which are postnatal day 10, intracranially with measles virus. And I was observing them daily for a period of 90 days to see how they are responding to the infection. Based off of my observation, I was able to develop a scoring rubric where a score of 0 was given to animals that are asymptomatic and a score of 5 was given to animals that died during the infection. So I have some pictures here to show as an example. So this was an infected mice that remained asymptomatic throughout. Very often the earliest sign of illness that I was able to see were ocular symptoms, where these animals had difficulty in opening their eyes because of crusty eyelids or pus in the eye. If you see here, the left eye shows ocular symptoms, right eye appears normal. Now some of the animals become more sick and they go on to develop ruffled fur, hunched posture, problems in movement before they become moribund, and ultimately they die during the infection. And what I was seeing is when these animals die, they have this typical posture of a hunched back and ruffled fur. So this scoring rubric also allowed me to develop a graph. So on this graph, on the y-axis we have the number of mice, and on the x-axis we have the days post-infection. The scoring rubric here is flipped, so at the top you have the dead score and the bottom you have score for asymptomatic mice. And what this graph told us was that animals that get a score of above 2 do not have the potential to recover and survive the infection. But when animals have a score of 1 or 2, they can recover and survive. While doing these studies, my goal was also to develop a survival curve, which is shown here. And in this graph, on the y-axis, we have % survival, and on the x-axis, I have days post-infection. The group in white here are control animals which were injected with PBS, and as expected, they did not become sick or they did not die during the infection. The group in red here are juvenile animals infected with a lower dose of the virus, which showed about 23% mortality. The group in green here are juvenile animals infected with higher dose of the virus, which showed about 75% mortality. And then we have the neonatal mice, which was a positive control because all of these mice died during the infection. So based off of these studies, I next selected the lower dose of the virus to be appropriate for conducting all of my other studies, mainly because it gave me a good balance between a group of animals that became sick as well as asymptomatic mice. And on the other hand, it gave me sufficient number of animals to carry out long-term studies to ask some of those questions as to why there are long-term consequences after a childhood infection. While doing this, I was also monitoring the weight of these animals, and what I saw was that my animals were clearly divided into 2 groups. Majority of them did not become sick, they did not die during the infection, and they also did not lose weight. The ones that became sick and died, showed in red here, they are the ones that lost weight, and I could clearly see that split between the 2 groups. So these characterization studies helped me develop my study timeline, as shown here, where day 0 is the day of infection, so they are postnatal day 10. Day 6 was when early signs of illness were visible, which was ocular symptoms. By 15 DPI, animals that became sick died. And beyond 15 DPI, all animals that remained were the surviving mice. So based off of this, I selected 2 different time points. One was 9 days post-infection. Now in this time point, we have some animals that are sick as well as some animals that are asymptomatic. And the other time point was 90 days post-infection. All we have here is a surviving mice, and they are now into their adulthood. And at both of these time points, I harvested the brain of the animals in order to ask the question about what was happening in their brain with respect to the immune response, the neural cells, as well as the virus. So I want to first start off by talking about the immune cell profile at these different time points. I started off by looking at the innate immune cells. The innate immune response is non-specific, so it cannot distinguish between different pathogens. And within this system, I was looking at macrophage microglia, which are the phagocytic cells, and their goal is to kill off infected cells, but at the same time, they can release several antiviral cytokines to control the infection. I next looked at the neutrophils, which are generally the first responders when you have a peripheral infection. They come into the site of infection and they release several cytokines and chemokines in order to get things in motion. They can themselves clear the virus by releasing porphyrins or by neutrophil extracellular traps. And finally, I looked at the natural killer cells or the NK cells, and as the name implies, they kill off infected cells. So all of the 3 innate immune cells that I looked at were significantly higher in the infected group shown in pink compared to the control group in blue. And by 90 DPI, all the innate immune cells had returned to baseline levels. So what this told me was that the immune response is reacting to the infection, and the innate immune cells coming to baseline indicates at least some level of viral control here. I next went on to look at the adaptive immune cells, and the adaptive immune response, unlike the innate immune response, is specific to a particular pathogen. So it can distinguish between viruses, between bacteria, or between any pathogen that comes into the body. Within the adaptive immune cells, I was looking at the CD4 T cells, which are helper T cells, and they are really the ones that orchestrate the immune response. They can potentiate actions of CD8 T cells as well as B cells. And on their own, they release several antiviral cytokines. One of them is interferon gamma, which is really important for viral control in the CNS. I next looked at the CD8 T cells, which are cytotoxic T cells or killer T cells, and their goal is to kill off infected cells by release of porphyrin and granzymes. And then finally, I looked at the B cells, and these are the antibody-producing cells. So they are tasked to produce antibodies that are specific to the virus. So all of the 3 innate adaptive immune cells were also higher after infection compared to the control group in blue. And by 90 days post-infection, only B cells were present in the brain. I just want to mention here that in the adults in the CD46 model, where we don't see CNS disease and the virus is very well controlled, the CD4 T cells, either along with CD8 T cells or the B cells, are really vital to get the virus under control. And the fact that they were present at the juvenile age told me that the immune system is responding with an idea of getting the virus under control. So overall, what we saw until now was that at an early time point, you have both the innate and adaptive immune cells coming in, despite illness. So illness did not seem to affect the ability of the immune cells to get into the brain. When the animals survived the infection, they still had B cells present in the brain. And the presence of these B cells in the surviving animals led me to further investigate more about the virus and specifically ask where the virus was present in the brain, what are the brain regions that contained it, and do these survivors actually control the virus or not? So I looked at the virus by a couple different methods. The first one that I was doing was flow cytometry, for which I have these graphs above. In these graphs on the y-axis, we are looking at the percentage of infected neurons, because neurons are the ones that are infected in our model. I'm also showing you only the hippocampus data, which is one of the brain regions that contains stem cells in the brain. So as expected, at the early time point, we saw that the virus was present in the brain. But surprisingly, even in the surviving animals, the virus was not controlled and we could still detect viral antigen. By 150 days post-infection, so this is 5 months after, the viral antigen levels had returned to baseline levels. And this tells us that the juvenile antiviral immune response ultimately does get the virus under control, but it takes it months for it to do so. And at this point, we don't really know at what exact stage the virus is controlled, but sometime between 90 to 150 dpi, the immune response is successful. The image here is an immunohistochemistry image of a mouse brain. The white arrows are pointing towards infected cells. I don't know if you guys can see this because of the light in the image. So the white arrows are pointing towards infected cells here, which are in red, and all other cells are in blue. And we can see that there are several regions of the brain that are infected at this time point. So I'm just going to zoom in into this circular image, which is shown here, and you can appreciate that both the cell bodies and the processes are stained with the virus. When I did immunohistochemistry in the brain at 90 dpi in the survivors, our IHC data corroborated with the flow data where we saw that the virus was still present in the hippocampus. But apart from the hippocampus, other regions of the brain like the cerebral nuclei also contain the viral antigen. And actually the cerebral nuclei was one of the hotspots for the virus to be present in the survivors. All the animals that I looked at did have virus in the cerebral nuclei. I do want to mention that although there are several regions of the brain that are infected even in the survivors, comparatively fewer number of regions contain the virus in the surviving animals compared to the ones early in infection. And that tells us that yes, the virus is on its way out and it is getting controlled, but it has just not happened yet. So overall, what we have seen up until now is that early during infection, you have a group of animals that are sick as well as that are not sick. And despite the sickness, both innate and adaptive immune cells are present in the brain. The viral antigen is widespread with multiple regions containing it. In the brain of the survivors, the B cells still remain and the virus is present. So perhaps the B cells are around to get the virus under control. So with this context, what we know here is that the immune cell profile is really different early during infection as compared to the later time point. And this led me to further ask my next question. How would this difference impact stem cells in the brain of these animals? And what happens to these stem cell activity? Before I go into that data, I just want to talk to you a little bit about stem cells in the postnatal brain. As I mentioned before, with age, the stem cell pool decreases and it becomes restricted to two specific areas. One of them is the hippocampus, where the stem cells are responsible to form glutamatergic dendritic neurons to assist in memory and cognition. And the other region is the subventricular zone, where the stem cells form neuroblasts that migrate out of the SVZ into the olfactory bulb shown in pink here. For the purpose of today's talk, I'm just going to be focused about the hippocampus for the rest of my talk today. So in order to mark stem cells, I used a marker called as nestin. Nestin is a gold standard in the field, and stem cells at all ages are marked by nestin. In this image here, I'm showing you embryonic stem cells in vitro, which are stained by nestin, shown in green. So using nestin, I did flow cytometry for the juvenile animals. And I saw that early during infection, there is a drastic decrease in the stem cell pool in the infected group in red compared to the control group in yellow. And in the brain of the survivors, by 90 dpi, this pool recovers and the numbers are comparable to the control group. So when I saw this decrease early on, it further led me to ask the question, why are these stem cells decreasing and what is happening to them? I looked at three different possibilities to answer this. One of them is that there is cellular death happening because of inflammation in the brain, and that leads to a decrease. While that was true, we did see modest cell death in the brain in multiple regions. it did not fully explain the extent of the decrease that I was getting here. So cell death was not massive at this time point. This further led me to ask my second question. Is it possible that stem cell proliferation has decreased because of inflammation? However, that wasn't the case either. Proliferation remained unaffected at this time point. So then I finally asked my third question. Are these stem cells moving out of the stem cell pool and forming other cells in the brain, like the neurons or the glial cells? And while they do that, NSCs exist in multiple different differentiation stages before they can commit to either forming a neuron or a glial cell. And in all of these different stages, they do express nestin as a marker. However, one of the limitations of my early study was that nestin is a broad NSC marker. It marks NSCs at all of these different stages and all of the subtypes. So one challenge that I was facing is I wasn't able to tell whether the inflammation in the brain is sensitive to specific NSC stages or does it lead to a massive decrease of the entire pool. So in order to get a better handle on that, I next devised a staining scheme based on the literature to segregate the different stages of NSCs. So undifferentiated NSCs were labeled with nestin and SOX2. Mid-stage NSCs were labeled with nestin and Musashi. And NSCs that are early in the neuronal differentiation pathway were labeled with nestin and DCX. While doing these studies, what I saw was that the undifferentiated NSCs, which are positive for both nestin and SOX2, they did not change after infection. But the mid-stage NSCs that are positive for nestin and Musashi decreased after infection, indicating that the early decrease in the nestin pool that I was seeing was coming from mid-stage NSCs and not NSCs that are undifferentiated. Further, looking at NSCs that are early in the neuronal differentiation pathway, so these are the stem cells that express both nestin and DCX. They have just started going down the neuronal differentiation pathway. This pool of cells also decreases after infection compared to the control group, leading me to conclude that the decrease in the nestin population was coming from mid-stage NSCs and NSCs that are early in the neuronal differentiation pathway, but not from undifferentiated NSCs. While doing these studies, I also saw that the immature neurons, which are cells that express DCX but do not express nestin, so these are the cells that are more closer to forming mature neurons, having lost the stem cell marker nestin. This pool of cells also increased after infection compared to the control group, indicating a push towards forming more neurons in the brain at this time. So overall, these studies led me to understand that the inflammatory milieu is sensitive to NSCs at specific stages. And what I think is happening, that the decrease in the stem cell pool is actually not because of they are dying or they are decreasing their proliferation. But it's mainly because they are moving out of the stem cell stage to form other cells like immature neurons here, as I did see this pool increase after infection. Now, as NSCs can move down the neuronal differentiation pathway, and I've spoken a lot about how they form immature neurons, there is a possibility that some of these cells can move down the glial differentiation pathway as well. So are they forming glia, and that may also contribute to the decrease in the nestin pool that I was seeing. I'm going to talk to you more about the oligodendrocyte progenitor cells, or OPCs. These cells mature to form oligodendrocytes that myelinate the central nervous system. And the reason I actually started looking at these cells was because myelination is an important physiological developmental process that is happening in these animals at this time. So I started investigating more about OPCs and how they are responding to the infection. Unlike the stem cells, the OPC pool increased early during infection, and this increase was maintained even in the brain of the survivors, where OPC increase was detected. And this tells us that you can survive an infection, and some of the cell types can return to normal, like NSCs did, but some other cell types, like OPCs, still remain high and are abnormal in the brain of the survivors. So long-term cellular changes do exist after a childhood infection. The increase in the OPC pool led me to further question the source of the increase. The reason I looked at this was that apart from NSCs, OPCs are another pool of cells that can proliferate a lot in the postnatal brain. And they maintain their proliferative property throughout the lifetime of the host. So I started looking at the possibility of increased proliferation after infection, but like NSCs, OPC proliferation also remained unchanged. So this brought me back to my same question that I have been asking as to can NSCs move down to form OPCs and does that contribute to this increase? So in order to look at this, I was doing flow cytometry where I was labeling stem cells with nestin, OPCs with O4. And my goal here was to look at this mid-stage of cells that are expressing both nestin and O4. So they have started going down the oligodendroglial lineage. I'm just going to pull up my timeline here for a bit because I did these studies at 2 different time points. One was 9 DPI, where we already know that the stem cell pool has decreased, the OPC pool is increased. And the other one was 5 DPI, which is early during infection. And I wanted to capture these cells before they had changes in the pool. So in these graphs here on the y-axis, we are looking at the number of NSCs that have started expressing O4. So we are looking at the nestin positive, O4 positive pool. And we see that there is an increase in this pool after infection shown in green, not only at 5 DPI, but also at 9 days post-infection, showing that NSCs are moving down the oligodendroglial lineage. So I knew that, okay, NSCs are coming down to form OPCs, but OPCs ultimately help in myelination of the brain. So my next question was to see if myelination was perturbed in any way, which is why the OPCs are responding with an increase. Before I go into the myelination data, I just want to talk to you a little bit about the myelin shield. Myelination is a process that happens both in the central and the peripheral nervous system. And in the CNS, the oligodendrocytes bring about myelination. So as you can see here, they extend their processes and they wrap around the axon in multiple concentric layers to form the myelin sheath. And this sheath is made up of lipids, proteins, and water. It is a highly lipophilic membrane, the majority of it is lipids. It also contains proteins, and myelin proteins are really the ones that maintain the integrity of the myelin sheath and compaction of the sheath. These proteins are divided into 2 groups, major myelin proteins and minor myelin proteins. The major ones are MBP and PLP, which are really responsible for compaction and integrity. And the minor myelin proteins are CMP, MOG, and MAG, all of which help in communication between an oligodendrocyte or an axon, and they also in some way maintain the integrity of the sheath. So when we think about myelination, the function that we all know about is that it helps in conduction of faster impulses through the CNS and the PNS. Because a myelinated neuron can do faster conduction as compared to an unmyelinated neuron. That is true, but apart from that, it also supports axons, metabolic support, and axonal insulation because of its high lipophilicity. And what has been seen is that in diseases of demyelination, where the myelin sheath is broken down and the axon is exposed to the extracellular environment, such axons die faster as compared to unmyelinated axons. The outcomes of demyelination really depend on the brain region that it is happening. So for example, in the hippocampus, if you would have demyelination, it can lead to deficits in memory and cognition. But if you would have demyelination in the motor cortex or the spinal cord, it leads to problems with movement and mobility. So with this context, I next looked at a possibility for myelin disruption after infection. So I started segregating the brain into 4 different regions. The first one was the hippocampus, which I've been talking about for all this while. The second region was the subventricular zone, which is another region in the brain that contains the stem cells. The third region was this marked with blue here, which is the cerebellum. And the remainder of the brain shown in green that I've traced here is the rest of the brain. So in all of these regions, I was looking at both the major myelin protein as well as the minor myelin proteins. The major ones were MBP and PLP, the minor ones are MOG and MAG. I'm going to start off by talking about the MBP first. MBP is a developmentally regulated protein. It has 13 different isoforms. and each of these isoforms are regulated with age. In this example plot here, you can see the red is the gap pH, which is the loading control, and all of the different bands for MVP are shown in green. So the way I analyzed this plot was that the higher molecular weight MVP was the top isoform. The mid-range for the molecular weights of MVP was the middle isoform, and the lowermost molecular weight of MVP was the bottom isoform. I also have a fourth graph here, which was all MVP isoforms together, which was total MVP. So as you see in these graphs, early during infection in the hippocampus, we do not see changes in MVP expression. But in the brain of the survivors, we see a significant decrease in the bottom isoform of MVP and a trend towards a decrease in all MVP isoforms. I also want to mention that each isoform of MVP has specific functions, all of which is not very well known, but the bottom isoforms of MVP are associated with remyelination. So decrease in these bottom isoforms leads me to question of whether these animals can perform remyelination well or not. And what we see here is that one of the major myelin proteins is altered in the brain of the survivors long term. I next looked at PLP, which is another major myelin protein. And very similar to MVP, even PLP did not change early during infection. But the brain of the survivors had a significant decrease in PLP expression, again telling us that both major myelin proteins are altered in the brain of the survivors. Finally, I looked at the two minor myelin proteins, which are MOG and MAD. And again, like MVP and PLP, even MOG and MAD did not change early during infection. But the brain of the survivors had a trend towards decrease in MAD expression. This leads me to conclude that in the hippocampus, you have both major myelin proteins decreasing, as well as a minor myelin protein decreasing, indicating towards a phenotype of demyelination in the brain of the survivors, at least with this brain region. Apart from myelin proteins, I also looked at myelin lipids, and I've stained this with fluoromyelin green here, and the green shows you fluoromyelin green. The top two images here are the hippocampal images. Now these are from the animals that are early in infection. So this is a control group, this is infected group at 9 dpi. And even though the hippocampus did not show alteration of myelin proteins, lipid staining decreased significantly. And I say significantly because I have quantified this data, and fluoromyelin green intensity does decrease, showing impairment in myelination even at the early time point. The bottom two images are of the corpus callosum, which is a highly myelinated area in the brain. This image here is after infection, and this is the control group. As you can see, the signal intensity does decrease in the brain of the survivors after infection. So with 3 months down the line, their myelination is not normal in these regions. So I just showed you data for the hippocampus today for the macromyelination. But I've also looked at the myelin proteins in the other regions of the brain. And early during infection, even though myelin proteins were not altered in the hippocampus, other regions of the brain did show alteration of myelin protein, telling us that myelination has started to disrupt very early in infection. In the brain of the survivors, also other regions did show a disruption in myelination. And no matter what brain region I looked at least one myelin protein was altered, indicating that whatever is happening is widespread, it's a long term, and these animals are not okay. So you can survive an infection, but the effects of it is not benign. There are cellular changes happening, developmental processes like myelination are really altered long term. So as myelination was disturbed in the brain of the survivors, along with an increase in the OPCs, it led me to further question what was happening to the mature oligodendrocytes, because they are the ones that ultimately bring about myelination. So is it possible that the mature oligodendrocytes are distorted in any way, which is why we have disruptions in myelination? So like any other cell type, even oligodendrocytes follow a very stage-wise differentiation pattern before they can form mature oligos. And at each of these different stages, they express a variety of proteins. So I took advantage of what was available in the literature to develop a staining panel for flow cytometry. And I've shown this here in this table here. OLIC2 is mentioned in yellow because OLIC2 is a pan-oligodendrocyte marker, so all oligos at any stage of differentiation should be expressing OLIC2. So in my flow cytometry data, I was specifically looking at the OLIC2 positive cells, and within that population, I was looking at the other markers. I can talk to you about the dating if you have questions in detail later. So the brain of the survivors, what I saw was that at least in the hippocampus, there was no change in the early OPC pool, but the intermediate OPCs, the late OPCs, and the mature oligodendrocytes increased in number, showing that there is a drive in the hippocampus to form more and more mature oligodendrocytes. And perhaps that's because demyelination is still ongoing in this region. So what we know is that the NSCs are moving down to the OPC lineage. The OPCs are also pushing forward to form more mature oligodendrocytes. But myelination is still abnormal in this time point. I don't exactly know what is happening there and why there is a disconnect between the two. It could be possible that the oligodendrocytes that are formed are dying and they're not able to survive, so they're not able to compensate for myelin damage. On the other hand, it is possible that these oligodendrocytes are doing myelination, but the extent of which is not enough to compensate for the damage that is being done. So just to conclude everything that I showed you today, what I saw was that after a juvenile CNS infection, there is an early time point where you have animals that are sick as well as animals that are asymptomatic. Despite that, you have virus present in multiple brain regions. The immune response is in full swing in the brain. This inflammation leads to a decrease in the stem cell pool. Now, the decrease can be in part due to the modest cell death happening, but I think it's majorly because the stem cells are moving out to form immature neurons, perhaps to compensate for neuronal damage as neurons are infected. But also they are differentiating to form OPCs. And by this time point, myelin disruption is already underway. So it's possible that the brain is getting a signal to form more OPCs to try and start compensating for the damage. Now, majority of the animals do survive the infection without overt sickness. And in these survivors, although stem cells have returned to normal levels, the OPC pool is still high. And there is more formation of mature oligodendrocytes in the brain, along with myelin disruption. This might all be an effort to repair damaged myelin. Now the question is the brain successful in bringing about myelin compensation after doing all of this and spending energy to form OPCs? I don't know the answer to that exactly, but over COVID, I have to thank COVID for this. I was able to keep some of my animals up until 150 days post-infection. And when I came back from the lockdown, I saw that 2 of these animals had seizures and abnormal gait, something that I had not seen at 90 dpi. And these neurological outcomes tell me that I think the brain has failed to remyelinate at some level, which is why you're seeing those overt outcomes in 150 days. And to remind you, by this time point, the virus is already gone, the inflammation has come down, and despite all of that, the brain is not succeeding to bring about compensation for myelin damage. So take-home message really is that juvenile neurotropic infections lead to enduring disruptions in myelination. That is long-term, long-lasting, and perhaps for the rest of the life of these animals, they are suffering from neurological outcomes. This was all that I showed you today, but I have done more work over the past 6 years, and I don't have time to talk about everything, but I'm just going to give you small snippets of what I have been doing other than looking at the hippocampus. So I also was able to look at the subventricular zone, which is another area of the brain that contains stem cells. And the impact in the subventricular zone is very similar to what we see in the hippocampus, at least with respect to NSCs, OPCs, and myelination. But more importantly, myelination was most severely damaged in the SVZ as compared to any regions of the brain. One of the functions of the stem cells in the SVZ is to move out of this area into the olfactory bulb to support olfactory neurogenesis. So I did look at olfactory neurogenesis as well, and I saw an increase early during infection, but in the brain of the survivors it had returned to normal levels. One of the things that I recently started doing was trying to look at immune factors that would help us understand why stem cells were showing the response that they are, what is impacting OPCs and myelination. So I started this by looking at interferon gamma, which is a crucial cytokine not only in this model, but other RNA viral infections of the brain. And I started infecting interferon gamma knockout animals, so these animals do not have interferon gamma. And I looked at them at 9 days post-infection. So by this time point, almost all of the animals are sick. All of them have seizures, something that I have not seen in such an extent in the wild type animals, telling me that the presence of interferon gamma is very important in this model. These animals also had a different immune cell profile, so greater CD8 T cells, lesser NK cells. There was more virus as compared to wild type animals. The impact on NSCs was actually opposite, so stem cells increased, but the impact on immature neurons and OPCs remained the same. In order to further understand the impact of the interferon gamma on stem cells, I was able to look at them in isolation in vitro. So I essentially grew embryonic stem cells in a dish and I treated them with interferon gamma. And what I saw was that there's a push towards forming glial cells other than neurons. So this in some part does recapitulate what I was seeing in vivo also, where there is more response to form oligodendrocytes. So this project has really opened up a lot of exciting avenues for the lab, and we have a lot of questions that we want to ask as a next step. Something that I have asked everywhere, I've been asked everywhere, and I'm sure a lot of you are thinking right now, is why do some animals become sick and some don't? And I've looked at my data with all possible angles and trying to segregate it with sickness, but really nothing stood out until I reached myelination. And what my preliminary data suggested was that sick animals have worse myelination. So really the question is, does demyelination or an impairment in myelination govern sickness? And if that is true, can we do something to improve myelination so that these animals are not sick and they can actually have a good life? I was really surprised with 150 DPI time point. I'm grateful. I know COVID was really bad for all of us, but I think for my project, COVID turned out to be really important. And because I saw the neurological outcomes at 150 DPI, I do want to look at the immune factors a little more closely. I looked at interferon gamma, but one of the things that I really want to do as a next step for this project is look at B cells. The reason I say that is because B cells are present in the brain up until 90 DPI. B cells have a lot of history in multiple sclerosis. They cause demyelination by producing myelin-specific antibodies. And B cells do play a role in OPC proliferation and differentiation. So that's one thing that we can start looking at as a part of the immune system. And then carefully more characterize the behavioral outcomes. What I looked was really by chance, it was serendipitous. I was not hoping to see any behavioral problems at 150 DPI. But trying to look more clearly into memory tests or olfactory tests would give us a better idea of the extent of impairment in these animals. Because hippocampus and subventricular zone govern memory and olfaction in some way. And both of these regions have abnormal cellular populations in the brain. And then finally, I think a practical question for us to ask is, these animals have survived the infection. What would happen if there is a second hit? If the virus comes in again in the brain, do they actually respond to the infection much better, or is there more brain damage that leads to a quick dropout of the animals? Yeah, so with that, I have a lot of people to thank because I don't think I could have done all of this without the support